time of 2D NMR and pulse sequences and so on. So that was very soon after my first book on NMR, which was published in 1986 by Springer. May I have the first slide, please? Where are my slides here? So please alert me 2 minutes before my time is up, because I don't want to take somebody else's time. Thank you.
This is my time, so don't worry.
Then I will give you 2 minutes of my time to you. Thank you, Iqbal. So my lecture will be a little bit about NMR. So the conference is about NMR, a little about what we are trying to do in this to uplift science, and then some of the disruptive innovations that we heard about this morning. So this is an 800 megahertz NMR in our institute, where much of the work with macromolecules is carried out. And you'll be having a lecture on saturation transfer difference NMR spectroscopy tomorrow from Dr. Athiya. She'll be talking about some of the work that's going on to look at mechanisms, etc. And this is one of a series of different NMR spectrometers. We also have a helium liquefaction facility. So any helium gas that's evaporated from the NMR is recollected into large balloons, recompressed and reliquefied. So there's a recycling facility, and these are some of the.
Excuse me, sir.
An audience of this type, I don't want to explain the basics of NMR. You all know about it.
Excuse me, sir. Sir, please share your slides for online participants.
Somebody's saying, please share your slides. Slide is not shared. Sorry, I beg your pardon. You just wants to share slides. Share screen. Just push the share screen. No, I should push that. Yeah, now you make it bigger. Can you see the slides now?
Yes, sir.
You can say yes, please, if you can.
Yes, sir.
Okay, good. There's a baby listening to my lecture. Very good. Screen. Yeah, okay. Let me see if we can change the slides now. Yeah. Okay, very good. All right, I'm just trying to make it bigger if we can. Yeah, that's it. Okay, good. So let me, these are now small tabletop NMR spectrometers, which are also available. They can go up to 90 megahertz, or they may be even bigger, perhaps 100 megahertz. And they don't have quite the sensitivity of the bigger instruments, but they're still very useful for some of the rapid benchtop work. For instance, this is an NMR spectrometer. Let me just close this off if we can. There is this thing blocking it. Can you switch that off? Yeah. Yeah, it's not very clear, but still, basically just showing that within a matter of 10 seconds, you can record a good NMR spectrometer using these benchtop instruments which have a permanent magnet. The slide is not changing now.
The slide is not changing.
Slide is not changing.
Yeah, can you?
We will do it. Oh.
Sorry,
It's okay.
let me, first of, it's possible. On the Zoom, yeah. Hide this.
Yeah.
I know it will come back again.
Yeah, okay.
Yes.
Okay, very good. So these are, you also have earth field NMR spectrometers where the earth is itself being used as a magnet. And this is very interesting. They have a very small proton frequency. They have some, they operate between 25,000 to 65,000 nanotesla, depending on where you are on the equator or in the poles. There is a significant difference of the magnetic fields. And you know, also have dynamic nuclear polarization, magic angle spinning NMRs, and especially for radicals, large electron polarization of radical molecules that is created can be transferred to a nuclear spin, which is of interest. And this can give you a significant enhancement of sensitivity. Nitrogen-cooled cryogenic probes are very common. So if you have a 400 megahertz, instead of buying a 600 or 800, which of course will give you a greater dispersion, but if you're looking for just for sensitivity, it's much more convenient just to go for a cryoprobe attachment, which will increase the sensitivity by 2 or 3-fold easily. So many of our instruments in Karachi are fitted with the cryoprobes. Dual receiver, this is of advantage. There are experiments like Pansy and Panacea, in which each receiver is tuned to a different nucleus, which means that you can record multidimensional NMR experiments more rapidly. So our 800 megahertz, in fact, is a dual receiver system in Karachi, which allows experiments to be done much more quickly. But let me just focus for a while here on what NMR can do in the area of biology and medicine. The 3D structure determination of proteins and nucleic acids, looking at the folding of proteins and their stability, the binding studies, and you'll have a lecture tomorrow on this, looking at how proteins or DNA are binding to various ligands, protein dynamics, and the type. It's possible to look at timescales varying from picoseconds to days, looking at structure of biomarkers and metabolites in real time. So you can inject a person and with a certain molecule, and as it metabolizes, you can connect that person through a tube with the NMR, so that it's a loop system where you yourself are connected up. And then as that compound breaks down, you can detect the metabolites if they are formed. Many of them may be transient and unstable. So you can detect them as they are formed and as they disappear. So these are, this is also possible. And of course, looking at biofluids and tissues and in vivo NMR, and in hospitals, MRI, magnetic resonance imaging is very common. And they have, they work between 0.5 to 3 Tesla. And here you're looking actually at water concentration of water molecules in tissues. So if you have a tumor, the amount of water is going to be much less than in a normal tissue. And so this contrast of water molecules. So in MRI, you're focusing just on water concentrations, and this the contrast is achieved through relaxation of different protons in different tissues. And the spatial information is provided by use of gradients. And these are some MRI structures obtained using this technology. Understanding of the function of the human brain and seeing how certain parts of the brain light up when a person is trying to think can be done through functional MRI. So here is a person trying to solve a problem, and then you see certain areas lighting up, And this is actually what you're looking at are changes in the blood oxygen levels. That's what you're actually looking at. It's actually a complex series of events that occurs because you have the cerebral blood flow and neuronal activation are connected. So when you start thinking, there is a neuronal firing, and glutamate starts to be released during the process of neuronal firing. And this glutamate affects the astrocytes, the cells which are nearby, causing changes of calcium ion concentration. That in turn releases nitric oxide, which is a vasodilator. So there is actually a series of events ranging from glutamate, calcium ion concentration, nitric oxide release, vasodilation, causing the arterioles to expand and draw more blood to provide the glucose needed by the neurons in the firing process. So it's all about, but ultimately, to cut this long story short, you're actually looking at changes in blood flow. That's what this is showing you. And you can also see that there is on the left side is a normal person thinking, on the right side is a person suffering from schizophrenia or schizophrenia. And there is a difference. You can see certain areas of lighting, certain areas of the brain lighting up in the case of normal patients, which are not lighting up in the case of patients suffering from certain mental diseases like schizophrenia. NMR has been considerably used for tissue metabolism studies. This metabolic information that is obtained reflects the way certain organs are functioning. And there are various methods available from which one can do high-speed imaging and collect both spectral and spatial information. And some of these are described in these various experiments. And these are available in a series of books, volume 1 of a series published by Bentham Science, edited by Professor Iqbal and myself. This was 6 volumes were published. So volume 1 of this describes, this is application of NMR spectroscopy. So this describes some of these techniques which are used for tissue metabolism studies. Similarly, if you have the muscle contraction and release, so if you take a carp, a fish, and if you expose it to water anoxic conditions where the oxygen amount is less, then it is deprived of oxygen. And so if you look at this phosphocreatine peak, which is the biggest peak here, under normal conditions, phosphocreatine is important in the ADP, ATP conversion, energy supply. And so if you under anoxic conditions, that is decreased. So you can see at the lower graph, the phosphocreatine, the large peak has become much smaller. And so this decrease is indicative. So these kind of studies of muscular contractions can also be done using NMR techniques. So you will be having several lectures over the next 2 days on structure elucidation, using various technologies. Here I thought I'll just give you a little bit of a flavor of some of the applications in biology and medicine. So you can also look at the effects on heating of oils, how they degrade as oils are heated. So do we have a pointer that I could sort of point? Is there a pointer available somewhere? Is somebody? Well, you can't see these slides very well here, I'm afraid. G. Thank you. You're a problem solver. Thank you. So basically the aldehyde protons, as you heat, an oil, the aldehyde protons come up. And so this is showing different types of oils, and the aldehyde protons are missing at the very bottom, and which is taken at room temperature. But when you heat it up, the amount of aldehyde protons, which basically is the amount of degradation products of the oil. So you can look at stability of oils, including the oils with which you cook food, how stable they are at different conditions, under different conditions. Looking at neurometabolites, the biomarkers for neuropsychiatric disorders, there are a series of biomarkers. And you can, each biomarker is characteristic for a certain type of disease if the concentrations of that biomarker vary from normality. So there are a series of these biomarkers which are available. And for instance, in the case of schizophrenia that we mentioned, the reduction of N-acetyl aspartate as a biomarker it tells you so that if this concentration of this particular biomarker is decreases, you see this happening in schizophrenic patients. So if a person comes in who has signs of schizophrenia, one could very easily check the level of N-acetyl aspartate, and if it is lower than normal, then it provides additional evidence that this indeed is schizophrenia. Similarly, in areas, various neurodegenerative diseases like autism, for instance, the choline-containing compounds and creatine, phosphocreatine are significantly increased. So increase from the normal of these biomarkers in autism is another very strong indicator that there's something wrong. In the case of bipolar disorders, you have, again, abnormalities in N-acetyl aspartate levels and choline levels. Similarly, in cancer studies, you find that there are higher levels of hydroxyisovalerate and some other compounds in the urine of lung cancer patients. So in fact, various types of cancers, there's a lot of very interesting work that has been done to detect certain metabolites and their levels and then relate them to various cancers. And there are a whole series of these in different types of cancers like leukemia or neck and bladder cancers. You see variations of different metabolites, which can provide you with concrete evidence that, yes, there is something wrong in this patient. Well, we have a number of books, some 13 books that we have written or edited, and Dr. Iqbal and I have together contributed to these books. My first book, in fact, was this one on NMR spectroscopy, which was published by Springer in 1986. And the reason I'm not an expert on NMR, I am a professor of organic chemistry, and my work is largely on synthesis and structure studies. But I wanted to go too deep into NMR and try to understand it. So the best way to try to go deep into an area is to try to write a book on it, because that forces you into a timeline that you have to submit this by this date. So in 8 months, 9 months, it must be of a quality which is acceptable internationally. And it also offers you an opportunity for learning. So I decided to start writing books on NMR if I wanted to learn NMR. So, but then I thought, how can I make a book which is different from those which have been written by experts on NMR spectroscopy? So all the books that were coming out at that time had the various pulse sequences described in the form of equations. So I said, can we describe this in the form of actual physical pictures of the behavior of magnetization vectors on application of different types of pulses? So then this entire book describes each NMR experiment with physical behavior of the magnetization vectors on application of pulses. And this became very popular and was translated into the Japanese version and so on. And this then led to my coming to Jordan and giving a series of lectures on all these, the behavior of magnetization vectors on application of different pulses. My second book on NMR, besides this, I decided also to write a, a chapter at the end, which described each of those pulse sequences in purely mathematical terms. So cozy, nosy, 2-digit resolve, head call, etc., etc. So the last chapter of this book is the product operator approach to NMR spectroscopy, which goes to the other extreme and describes each of the experiments purely in mathematical terms. So these two books provide you with a very interesting landscape of NMR, one providing a physical picture and the other providing a mathematical picture of NMR. And then Dr. Iqbal and I wrote this book, which was solving problems with NMR spectroscopy. And this was then revised with Dr. Atia joining us, and a revised version was published in 2015, and it's being revised again now. And we hope to have another revised version published by Academic Press in the next year or so. And these are other books on NMR. There are 6 volumes these are all on applications of NMR spectroscopy in different fields. So you may be interested in this series. Well, let me now switch to science and technology with emphasis on the emerging technologies. Professor Iqbal in the talk this morning mentioned the McKinsey Global Report of 2019, which emphasized the impact that these technologies will have in the future. And according to this report, there will be a $33 trillion impact of some of the disruptive innovations, including advanced genomics, industrial biotechnology, artificial intelligence, 3D storage printing, etc., by 2025, so very soon. So these are the so-called disruptive innovations, which are going to impact our lives in huge ways. And you can also already see how combustion cars are slowly becoming obsolete, and very soon you will have electric cars where you will need just one charge in 2 minutes. Instead of petrol, you charge your batteries for 2 minutes and it'll take you 1,000 kilometers. It's happening right now. This battery has not, the prototypes are already there. Commercialization is not, but this is going to happen very soon. 2-minute charge, 1,000 kilometers on a 2-minute charge. No petrol, no combustion engine, hardly any moving parts, just to give you an example. So in many ways, the world is changing. And so what I described this morning about knowledge being the key driver for socioeconomic development. So it's countries that have realized are investing in education, science, technology, and innovation are moving forward rapidly. Never before in the history of mankind have so many been transformed so quickly as has happened in China in the last 3 decades. And how did that happen? Last year, they sent 600,000 students for PhD and postdoctoral training abroad. Last year, 500,000 students returned back to China after getting their PhDs or postdoctoral training. So 500,000 highly trained professionals are being added to the Chinese workforce every year. They are being clustered in centers of excellence, and they are driving the dynamo, which is moving China forward, beating the United States in many fields, including nanotechnology, including the number of international patents granted, et cetera, et cetera, et cetera. So that is what countries like Singapore have done, and so on. And that is a path for countries like Pakistan and Jordan also. And let me move to the last section of my talk, the biotechnology and the disruptive innovation part. It takes thousands of years for new species to evolve. But now, using gene editing technologies, such as CRISPR-Cas, you can slice a part of a gene and insert it into another species and create new species within a matter of weeks or months. So the whole process of evolution can now be short-circuited by gene editing technologies. And the 2020 Nobel Prizes were shared by Jennifer Doudna and Emmanuel Charpentier based on their work of developing this tool, this molecular scissors, which can slice your part of your DNA. And there are new variations are coming out. This was just published this is a much softer version and a much more accurate version, which can deliver genes as long as 36,000 base pairs into human cells. And it is softer because it doesn't slice both strands of the DNA at the same time. It slices one and then slices the other sequentially. It's called PASTE, P-A-S-T-E. So this was published in Nature Biotechnology in November last year. So you may want to look it up, and there are every month or every 2 or 3 months, there are new developments that are taking place of such technologies. Similarly, the technology has been developed for personalized medicine and to redirect the immune cells to recognize mutations in the patient's own cancer cells, to triggering your own immune cells to fight cancer through such CRISPR-engineered immune cells. This was also published just 3 or 4 months ago. So this field is really exploding, and it's finding applications in many diseases, including cancer. Understanding aging. How do we get old? There's a lot of literature about the factors responsible for aging, whether it is programmed cell death, apoptosis, or the telomere, which degrades. And so there are substances now available which would allow the repair of that telomere, and which will allow, through activation of the enzyme telomerase, the aging genes have been identified in oxygen radicals. So aging is a chemical process, and many chemical processes are reversible. Can I or Dr. Musa be 17 years old again? And the answer is, theoretically, it's possible. And it has been shown that in animals, you can actually reverse the ages and make them younger. So when certain compounds, like resveratrol, were given to a 4-year-old mice, their body features changed, their behavior changed, and they became like they were 1-year-old. It has been done in animals. Nicotinamide adenine dinucleotide, NAD, is another, which has a strong anti-aging effect. A commonly used drug, metformin, in diabetes also has anti-aging properties, and rapamycin, and there are other compounds. The list is growing. So it's thought that children being born today will have ages of 120 or 150 years plus. So this is actually happening. So truth is indeed stranger than fiction. You've heard of 3D printing. Parts of human kidneys or livers or other parts of the body can now be produced by 3D printing. It's like an inkjet printer. And inside in a tank where you multiply cells, the patient's own kidney cells being multiplied and sprayed on layer by layer into the right form, in the right shape. And then these parts of organs can then be transplanted into the patient so that his damaged kidney can be repaired with his own 3D printed kidney cells. Amazing science. Neuroscience. This is perhaps the most exciting area of science that we know today. 100 billion neurons, each talking to 10,000 other neurons. 100 billion times 10,000 synaptic connections. Arguably the most complex object of our universe, our own brain. Understanding the functioning of that brain is perhaps one of the most challenging holy grails of science today. So that's an area where Professor Iqbal and I have been involved together for the last 2 decades now. So about 20 years ago, I published a theory at that time that thoughts are made up of certain molecules. And we proposed that the glycoproteins in the hypothalamus of the human brain, glycoproteins. What are glycoproteins? A peptide backbone with glucose, arabinose, fructose, xylose, etc. stuck on, bristling with hydroxyl groups, like a natural velvet that Allah subhanahu wa ta'ala has created in the brain. It's a natural velvet. And the ladies here would have worn embossed velvets, where you can emboss a pattern on a velvet. A similar embossing can be done in the brain. When I look at this microphone, a certain pattern immediately comes to my brain. So I had proposed that hydrogen bonding in the glycoproteins, through that process, the conformational mobility of sugars can be frozen. Normally, these are waving around, but that mobility can be frozen and folded proteins can be formed, and these folded proteins are the basis for thought storage. So that was and then so these patterns, then whatever you know, whatever you're learning, so learning becomes the formation of these folded protein patterns. Forgetting is the breakage of these patterns. And misfolding can cause problems like Alzheimer's and Parkinson's and other diseases might be due to the misfolding of some of these proteins. Or some of the assisting proteins, which are called chaperone proteins, which assist in the process of folding. So it's a very, very fascinating area. And these were published a long time ago, these kind of patterns of how intramolecular or intermolecular hydrogen bonds could have been formed. And so logic then becomes a multidimensional overlay of these patterns, the ability to think and resolve things. So this was a hypothesis. There's still an unproved hypothesis, I must say. I may be talking absolute nonsense just now, but there is growing evidence that I may have been right. And well, there are numerous reviews published in that, but just very recently, a few months ago, there was this paper which showed that if proteins are misfolded, then it causes major problems in the lasting forms of memory and in synaptic plasticity. So the folding of proteins in the process of learning and the misfolding of proteins in the process of various neurodegenerative diseases may well be true. There are many papers now which are supporting this hypothesis. Moving on, we were talking of the brain. So here is a man driving a car just by thought control. He's wearing a device in his head, and now the blind can see with their tongue. It's a lollipop-like device in the hand of this lady, which has 400 sensors on it. The moment she puts it on her tongue, the image, which is taken in the small camera on her glasses, is passed through her tongue to the brain, restoring partial eyesight. So you actually see with your brain. You don't see, you actually see with your brain, not with your eyes. And this is, so there are now different mechanisms available for restoring of at least partial eyesight.
Sorry,
Yeah, 2 minutes.
well, we can give you another few minutes.
Yeah, sure. No problem. I'll just wind up. So metamaterials have been developed, which have the ability to move objects, make objects disappear. So if I was to cover Dr. Musa Nazir with a shawl made out of a metamaterial, he is going to disappear. There'll be an empty chair sitting here. These are commercially available. They are being used for cloaking submarines and tanks because they have this amazing ability of bending light. So the light will hit you, it'll go around you and then hit the chair. So you will not be visible then. You'll become invisible. So Harry Potter's disappearing cloak is now a reality. So there are intelligent materials, self-healing materials, graphene, some 200 times stronger than steel. So these are the disruptive innovations which are transforming our lives. Some of them we don't know about till they come into the market. And those are the areas, the new and emerging technologies that we should be investing in. 3D printed jet engines complete jet engine produced through 3D printing, ladies' dresses, electronic textiles. You just press a switch and the design changes. So the husband don't need to buy new dresses for their wives anymore. They say, go and press a button. And these are ebb textiles which slowly change their color over 2 hours. And I can go on, but I think I will just stop here. Since my time is up and I don't want to steal Iqbal's time. So I'll just say the wondrous world of NMR spectroscopy is finding many, many applications in medicine, in biology, in chemistry, of course. And then the disruptive innovations that are taking place are changing our lives. And we need to train our children for the universities of tomorrow, not of today. I'll just show you one slide here, which in my talk this morning, I mentioned how the landscape of higher education and scientific research changed. Pakistan is green, India is red. International publications per 10 million population. This has changed and the green has gone up in 2017 and we are now about the same or a little ahead of India. This was no small achievement and it was done because of the measures that we took this morning. And those of you who want to read more about it, there is a book which came out a few months ago called Fostering Institutional Development and Vital Change in Africa and Asia by Fred Hayward, Palgrave Macmillan. So this is an entire chapter of what measures were taken and how the landscape of higher education changed. So thank you very much. And I hope, I wish you all the best. Thank you. Assalamu alaikum. If you want me to answer any questions, I can try. If there is time, I don't know.
Just want to thank you very much. You have taken us through a very exciting visit to the area of applications of NMR, applications of great importance. You have put people on the front line of research to enable them to see their future in the field. It's very exciting and really enjoyable to hear about these things from the master, from the founder in the field, from someone who has laid the ground for the applications. We thank you very much. And it has been a privilege to listen to you.
Thank you.
Thank you.
Now let's move on to the next speaker. And as you know, it is really extremely difficult for me to introduce a friend and an honest and dedicated person. So, excuse me, but I will be very brief in introducing you. I know Iqbal and I knew him in 1989 when he was young, and he's still young. When we have the Professor Adal in a conference in Karachi. Now it's a great pleasure to introduce one of the most prominent scientists in the field of natural products and bioorganic chemistry. Its contribution includes writing and editing 90 books published in USA and Europe. He has supervised about 100 PhD students, even more. And he has authored more than 1,200 research papers and 40 chapters, as well as 65 US papers. He has been among the most cited scientists of Pakistan. Now we are talking about citations. People say, I have 200, 2,000, and 3,000, and so on. That has more than 36,768 and more citations with an H index of 8. He is the volume editor of many international volumes. And this morning he was introduced to be the COMSTECH and and all these things. So I will stop here. Thank you very much. And he will be talking about science and technology, capacity building, contribution of the OIC, COMSTECH, and the ICCBS. At least he will tell us what is ICCB and what is welcome.
Excellency, my dear brother, Professor Dr. Musa Nazir, Professor Dr. Salim Sabri, very distinguished guests, my dear teacher, Professor Dr. Atar Rahman, guests from other countries, Assalamu alaikum wa rahmatullahi wa barakatuh. It is very difficult to speak after my teacher, Professor Dr. Atar Rahman. It's just extremely difficult because, you know, I joined him in 1983 when I used to have long hairs. And I used to wear jeans, which was often torn in different places.
I remember.
So, so he shaped me what I am today. You know, that is the touch of an angel. You know, and certainly his contributions in different spheres, including making people what they are, is absolutely phenomenal. So, thank you very much, sir. His last slide was about fostering institutional development was extremely important, and that is what I intend to introduce today. My work is very simple because I'm going to talk about two institutions, both institutions in which Professor Atar Rahman has played central role in the establishment. You can see several buildings. These buildings are on the basis of vision and dreams. And what they are and what they are contributing, I'll just speak about that. But before that, I want to introduce you, just for the sake of completion, that the Muslim world is actually have tremendous opportunities and daunting challenges. 1.9 billion population, the fastest growth of population in any religious ethnic group. Tremendous natural and human resources. So we are no dearth of natural resources or human capital. Over 50% of our population is below the age of 23. And with the using the words of Professor Atta, it is the tremendous pool of creativity which we have with us. Among 57 states, unfortunately, we have 21 of them are least developed countries. So the challenges are daunting. The gross expenditure on research and development is only 0.43 versus 1.78 of the world average. You know, so we're talking about countries spending 7% and above. Korea is spending 7%. On research and development, and then we have 0.43 as an average, including about 2%, which China with Malaysia and Turkey spending. One of the most important indicators, which really is an impediment in scientific development in the Muslim world, is participation, the lack of participation of women. 649 versus 2,532 of the world average. We are not really benefiting from this tremendous pool of excellence which we have in our bright, bold, and beautiful ladies. Muslim world faces enduring challenges of population bulge, youth unemployment, food, energy, water insecurity, and impact of climate change. But more specifically, we also have challenges such as regional conflicts, bad governance, illiteracy, lack of women participation, religious and cultural intolerance, and of course, Islamophobia, which we all face today. See, science can solve in solving many of these issues and challenges. We have seen green revolution. Now we have precision agriculture and biotechnology feeding the hungry. Sustainable agriculture can ensure that there will be no famine in the world of tomorrow and today. Global life expectancy exceeds to 76 years. And as Professor Tarman mentioned, it's going to be 100 years. So many of you are going to spend productive and healthy life to the age of 100 and even more. Antibiotics and antivirals and many of these tremendous development in the field of biomedical sciences. We are now living in a world where we can have tremendous benefit from these development. And of course, modern education technologies can ensure a global education for all. Now, as the last slide of Professor Atta is about fostering institutional challenges, because without a strong institution, be political institution, judicial institution, lawmaking institution, education institution, R&D institution, you need to have good institution, strong, objective institution, and without that, you cannot really develop. And when we talk about science and technology, you need to have R&D institution, you need to have a science support institution, you need to have very high quality education institutions from school to universities. And that is the greatest challenge which Muslim world face, the weak institutions. I'm going to introduce you to one institution, which sits right in the center of science and technology in the Muslim world. And work is one of the OIC institutions, is the OIC Ministerial Standing Committee on Scientific and Technological Cooperation. This beautiful building is right on the Constitution Avenue in front of Prime Minister's Secretariat in Pakistan. And this was built during the 16-year tenure of Professor Dr. Tarman. It's an iconic building. It's called 57 Flag Building. I invite you to come. This is a beautiful structure in which we are trying to do lots of good things despite many constraints. So it was established in 1981 as an institution and is chaired by the President of Pakistan and co-chaired by the Prime Minister of Pakistan. So very high level of political ownership, which we have for COMSTECH. And this is the organizational structure. So we have General Assembly, which is comprised of all 57 ministers, including your Minister of Higher Education and Scientific Research. Underneath, we have Executive Committee of 9 ministers, including the Minister, as well as Secretary General of OIC, chaired by the President of Pakistan, which takes immediate decision. And I'm there right at the bottom as a Coordinator General. And of course, there is a General Secretariat, which you have seen. Okay, so the aims and objectives of COMSTECH in very few words: assess what we have. We serve as an observatory of science and technology development in the Muslim world. So theoretically, for all 57 countries, every year, we monitor the progress in terms of education, science, technology, innovation, science park, patent for all 57 countries. Of course, it's not possible to get the data for all countries, but we try our best. And for every country, every year, there is a report, and it's a much thorough report than UNESCO Science Report. We also determine the country-specific needs, and I'm going to tell you what that means in terms of various programs of COMSTECH. And also we make sure that we develop the capacity of not only countries, institutions, but also individuals. This is the COMSTECH General Assembly. So it's a huge affair, So literally it's like a UN General Assembly where we have many ministers coming, Secretary General of OIC coming, ASG of OIC coming, President IDB is part of it, observer states. We have many observer states. Then we have recently, as has been mentioned by Excellency Askar Musanov, we have initiated OIC 15 dialogue platform, which is going to be part of General Assembly as well. So many UN agencies are part of COMSTECH General Assembly. So you can see vertically and horizontally. Vertically, we work closely with OIC General Secretariat, with the Standing Committee on Economics and Commerce, SSCO in Amman, Islamic Organization for Security in Kazakhstan, SESRIC, which is a specialized organization of statistics and research in Turkey, OIC CERT, which is in Malaysia, and Islamic Academy of Sciences or Islamic World Academy of Sciences, which is one of the institutions of COMSTECH. And of course, horizontally, we have established many institutions. I want you to point out that two successful institutions of the COMSTECH in Verdun, Inter-Islamic Network on Water Research and Development, and Islamic World Academy of Sciences are actually located in Jordan. So Jordan plays an extremely important role in many of these science and technology, policy making, and other aspects of S&T development in COMSTECH and OIC. And COMSTECH major program, I'll just take you through very quickly. We have the largest research training program in the entire OIC system, 700 fellowships. We are spending about $1 million on fellowships and related activities. COMSTECH Health and S&T program, which is a very large program, for which we have very strong support from Islamic Development Bank, OIC General Secretariat, and also many of the organizations. COMSTECH Research Support Program, and I hope that you would be able to benefit from that with the International Foundation of Science, the World Academy of Sciences, UNESCO, International Center for Genetic Engineering and Biotechnology, and COMSTECH Consortium of Excellence, and now University of Jordan is part of it. COMSTECH country-specific program include Palestine. We have a 500 fellowship for Palestinians, for Yemen in public health, Nigeria in food security, Sudan in health, and Somalia in food security and health. COMSTECH Forum on Environment and Ecosystem is one of the flagship programs of COMSTECH through which we are working in Sahel region. Of course, we have legacy programs such as COMSTECH Awards, which recognize the best scientific talent in the Muslim world. We prepare OIC profiles, which I mentioned, COMSTECH Research Grants Program, Research Publication Program, COMSTECH Library Program, and then OIC 15 Dialogue Platform, which is a recent initiative. Now, I want to tell you about the fellowship opportunities, because that is what I think many of you will be able to benefit. I had a very good discussion with the minister, and we have agreed that COMSTECH will set aside $50,000, and Ministry of Higher Education and Scientific Research will set aside $50,000 in Jordan for supporting the mobility of Jordanian scholars and students to different countries, including Pakistan. So 500 scholarship and fellowship through PICA, Palestinian International Cooperation Agency, in COMSTECH Consortium of Excellence Universities. Virology and Vaccine Training Program through Biopharma, Indonesia, and recently Pasteur Institute in Iran. Fellowship for Muslim minorities. So COMSTECH is the only organization which is mandated to work with the observer state and also Muslim minorities in non-OIC countries. So we have initiated program with Ethiopia, with Sri Lanka. We are starting program, my colleague is here, we are starting program with the Mombasa in Kenya and many other. Fellowship for technician training program. So if you have anyone who wish to learn various different kinds of instrumental operation, troubleshooting in the field of medical sciences, biomedical devices, chemistry, physics, mathematics, please do let us know, and we'll be able to help you to get training in various technology-related technician training. We have special fellowship program for least developed Africa. So there is we set aside a substantial part of fellowships for least developed countries, and of course, fellowship for women researchers. If you apply, you would get a fellowship because there is a sizable number of fellowships which are available for bright young girls. Another very important program which I would like to take your attract your attention is called Science in Exile. You see, the Muslim world has 5 recognized conflict zones: Yemen, Iraq, Syria, Libya, and I think Afghanistan. So 5 conflict zones, recognized conflict zones. Of course, there are plenty of them. And these 5 conflict zones that people have actually moved, migrated to other countries, and these are refugee scientists. You can benefit from that. Because of any reason, people have to leave their countries. You would be able to benefit that, and this is a flagship program with UNESCO and the World Academy of Sciences. Biotechnology Research Initiative with huge organization, ICGB, with 3 centers in Durban, in Trieste, and in India, in Delhi. Capacity building program, COMSTECH. We have institutional linkages. So through institutional linkages, what we were able to achieve, we developed the first faculty of computer science in University of Gambia. We are developing a halal, internationally accredited halal testing laboratory in IUIU, Islamic University in Uganda. We have transferred the technology of citrus processing to Nigeria through Arid Agriculture University, and all these 3 programs were funded by Islamic Development Bank. We're also working closely with Sudan for the establishment of National Center of Research. And of course, we have Youth Forum. So Pakistan, Turkey, Kazakhstan, now Saudi Arabia is also part of it. The second Youth Forum on Biotechnology will be held in Astana in May, and many of these programs which we have initiated. So there is a digital skill program, 5,000 digital fellowships are available, which we have initiated with some universities in Pakistan, and further universities are added into it. COMSTECH top priority is actually Africa, and here we really want Jordan to play an extremely important role. Jordan, both in terms of proximity and excellence, is an ideal place. You can help LDC Africa, and we can bring these people to Jordan for training, capacity building, and education. And that is what I wanted to request. So in Health Africa program, we have with many institutions, we are working in different countries. We had 2 camps in Niger. This month, we're going to have a chart on ophthalmology, which not only include retinal and cataract surgery, but also train doctors and select some of these doctors for postgraduate training in Pakistan through Pakistan Medical Commission. We have many campaigns for this year, including Uganda. We also work on repair and maintenance of biomedical devices with the help of partner institution, and of course, training workshop of different kinds. The Health Africa program also includes training in cancer care, Burkina Faso, Niger, and Sudan, and training workshop in neurology and neurocritical care. You know, this trauma and the stroke, how can one handle that at the emergency setup? This is Senegal, Guinea-Bissau, in Uganda. S&T education and research institution building in Africa, establishment of MS. I already mentioned some of these programs, and this is the partner institution. So in May, we are organizing a huge summit of 19 countries in Sahel region, which is going to be focused on environmental and ecosystem restoration. And this is going to be in Niger, Niamey. And COMSTECH Preventing Food Crisis in Sahel. This is an event which we are organizing in July. We also have a food security fellowship program, and many more of that. I'll just go very quickly. This is OIC COMSTECH technology portal. So if you have developed any technology which you would like to showcase, this is an official portal which COMSTECH operate for the OIC countries. And we also celebrate the great Muslim contributions. We had Al-Farabi Forum with the government of Kazakhstan in 2020. We had Iqbal Forum in Almaty. We're going to have a Babar Forum in Bishkek, and we're going to have Al-Baruni Forum in Tashkent this year. These are COMSTECH program for 2023. I'll provide you a list of them, but let me just take you through that. We have a very strong support from the government. Our ex-Prime Minister Imran Khan has visited our center, our secretariat twice. The president is a very frequent visitor of COMSTECH, and he has been supporting our initiatives at different levels. Of course, we have had the honor of welcoming the Secretary General of OIC. We also had a very detailed meeting. We hosted the President of Islamic Development Bank, and that has played an extremely important role in initiating many projects. And if you can see here, he is the foreign minister of Palestine who visited Islamabad and met with us. I also introduced you to a center which is very close to our heart, because 3 generations of leaders, 2 leaders, 1 follower, Professor Dr. Salim al-Siddiqui, Professor Atar Hafan, and myself. Since 2002, we were able to establish this institute. Was established in 1966, and this is now among the finest research institutions. So started from 2 laboratories or 3 laboratories in old chemistry building in 1966 with fine people, Professor Siddiqui, Professor Atar Rahman, Professor Waqaruddin Ahmad, and later Professor Zaidi also joined. These 4 people together have established a seat of this genesis of this wonderful institution. And now this is one of the largest research establishments in the developing world, with 17 research centers, with overall investment of $150 million only in the equipments. So you see a whole range of different institutions. I'll just want you to recognize the contributions of great people. So starting from Professor Salim al-Siddiqui, Professor Atta joined him in 1971. Mr. Aziz Latif Jamal was the first the donation which came to the institution for the establishment, and this was the largest donation at that time in the history of Pakistan. Nadra Panjwani, who in the memory of her fathers, Mr. Hussain Panjwani, has established a center. And of course, the 2nd and 3rd generation of these families are now helping us. This is the organogram of the institute. So university sits there. We remain by choice part of the university, ICCPS and 17 center in 2 broad areas, biochemical sciences and biomedical sciences. And these fascinating buildings, these buildings, each one of them has our heart in it, because every brick of these buildings, every brick of it is just so connected to what we were able to do. So a famous HEJ Research Institute of Chemistry, roughly about 3,000 square feet of laboratory space, huge lab, no offices, everyone. Professor Atta sit in his own lab, I sit in my own lab, every one of us, a great tradition. Then this is Professor Atar Rahman Laboratory Building. This is LEJ Nanotechnology Center, a specialized institution. We are spending about $1 billion rupees on this, and this has one of the finest material testing laboratory. We have Sino-Pakistan Center for Hybrid Research, Biotechnology. This is in the memory of late Professor Wolfgang Folter, a great man who has played an extremely important role in the establishment of the center. This is our LEJ Nanotechnology Center, LEJ National Science Information Center, which is connected to Pakistan Education and Research Network. This is UNESCO building, and this is the IEC, or Industrial Ethical Center. Similarly, in the field of biomedical sciences and biological sciences, we have several centers, including a very high quality genomic center, internationally accredited bioequivalence center, clinical research and hospital, National Institute of Virology, the Dr. Purvani Center, Synth Forensic DNA Laboratory, and many more, including National Facility for Laboratory Animal Research. So just take you through this institution is the most recognized institution in the Muslim world. It's the OIC Center of Excellence in Chemical Sciences. It is a UNESCO Category 2 center. This is this hosts UNESCO Chair in Medicinal and Bioorganic Chemistry. It is the World Academy of Sciences Center, Central and South African, South Asian Regional Center, WHO Center for Pesticide Analysis. It has received Islamic Development Bank Prize for Best Institution 2004 and 2010, and is also the World Academy of Sciences Center of Excellence for Training of Scholars. We are different, and I don't have time to tell you, but we have been different right from the beginning. So genesis of this center based on this great institutional memories which great people brought with them, and they made this institution very, very different. You know, so we think differently, we work differently, we work in a very different, with a different objective, with absolute sincerity of purpose. We had contractual appointment of faculty right from day one, from 1966. We have each one of us, including me, are evaluated every year against KPIs. And if you look into our KPIs, they are brute, you know. So we don't survive, each one of us from assistant professor to professor, 8+1, 9 evaluation. And if you miss one evaluation, you go out. So 9 evaluations. And then we have very high quality engineering staff who are capable of running some of these finest instruments in the world very, very efficiently. 60% of our fund goes to research and development, and only 40 or less we spend on salaries. So we make sure that we spend money where it is needed. We support young faculty members. So pyramid is reverse. We have more grant for young faculty members than the senior faculty members. We have for each of our students, we have a co-supervision. So all 600 PhD and master students are co-supervised by foreign experts, and we will request you to kindly be part of this very important scheme, which help us to improve the quality of quality of research. And each of the students, everyone who is admitted on the basis of merit, and it's only merit, each one of them is supported by fellowship. So we don't really ask them to pay any fee. We support each one of them. You know, every one of them is supported by scholarship, and they are quite generous. And of course, we have strong international linkages. Faculty with the stringent criteria, we judge them every year for citation, research output, international patent, book published, PhD output, international honors win, the national and international funds. We had, until recently, we had this wonderful scheme that if you bring ₹1 million, we'll give you ₹1 million. If you bring ₹0, we'll give you ₹0. So betting on the winning horses, and of course, we have been working closely with the government and with the society to serve them in many ways. Very high quality technical manpower. As I mentioned, very well-trained engineering staff who are capable of running these facilities successfully. And all students are selected from anywhere. So without any distinction of caste, creed, and nationalities, you can come and you can become part of the student body of this institution. And of course, we have world-class infrastructure. So we have state-of-the-art facilities in many of these areas, such as NMR. We have 20 mass spectrometer, and we'll be working closely with the industries also. And it is a very recognized institution. So we have been able to get out of 5 fellows of Royal Society, 2 are in our institute, Professor Siddiqui and Professor Atta. Mustafa, peace be upon him, Prize 2021, UNESCO Science Prize, and many of these prizes which we were able to, and also the Presidential Award. So this is the most celebrated institution in the country. And of course, many international awards and honors, including about 10 DSCs. So out of 70 faculty members, a relatively small faculty, 70 faculty members, 32 of them have civil awards. So you can imagine that it is actually a very high quality faculty which we have. This is a very historical moment when our center became the UNESCO Center. These are the historical occasions in which Professor Ataur Rahman has received UNESCO Science Prize, International Chinese International Friendship Award, and Chinese Award for Science and Technology from the President and Prime Ministers of China. This is when I received Mustafa, peace be upon him, prize. This is very unlike of me, a statue in somewhere in Tehran, which I have not really seen. And these are the same prize which I also received later this year, and also some further awards also. So this institution has been very productive in terms of research publications, patents, MPhil and PhD produced, citations and others. And we were also able to, right from the beginning, our founding fathers have worked very hard to get international grant. So right from 1966, this institution was built through philanthropic contributions, through international grants, and then later the government grants. And as a result of it, what has emerged is certainly something very interesting and important. We have very active collaboration with many institutions of the world. We are the home of 5 Sino-Pakistan centers. And of course, we have done in our internationally accredited triple recognition. We are a contractual research organization, we are a clinical trial unit, and we are also Bioclin Center. We have conducted literally several dozens clinical trials on pharmaceutical, including Sinopharm. And there are lots of international linkages, key capabilities in synthesis and analysis, instrumentation, and capacity building. And we also have done many things. There are some of these major breakthroughs, which I don't want to get into it because so my colleagues are going to talk about that. And is accredited from almost all forums. This is a biosafety and biorisk research capacity for the country. We have BSL-3, 5,600 square feet of BSL-3. We are in the process of planning BSL-4 for the first time in the country. These are BSL-3, which is homemade, it was all manufactured in Pakistan. So state-of-the-art, very high quality biosafety level 3. We have been very active during COVID and conducted lots of seminal work, including several clinical trials, genome surveillance, variant detection, and about 500,000 PCR tests. We are also official laboratory for our province. So we are SIN forensic DNA and serology laboratory, food testing lab, drug testing lab, and many more for the province of Sindh, which is the second largest province in the country. We have been supporting industries and generating roughly about 30% of our funds from services to the industry. And of course, most profoundly, I think this center, right from the inception, is very active in global capacity building, despite the fact that we are part of, we are in a developing country with constrained by number of factors. We have been working very, very hard to make sure that we contribute in global capacity building. These are some of the scholarship schemes available, but if you do not really find any, just contact us and we'll make sure that you get one. And you'll be able to, just finishing, sir, just last slides. And these are foreign students from different countries.
We have a few minutes to finish, please.
Less than few minutes, sir. About a minute. And these are German scholars. This is the only institution where we used to have German scholars. The former president of Mauritius, she was our alumnus and she was a postdoc in 1997. When she became the president of Mauritius, she decided to visit Pakistan, the first country, first stopover in Karachi, first activity to this institute. So we have a very strong international footprint. These are various institutions which are built in different countries of the world in recognition of our contribution. This is Professor Taurman Center for Natural Product Development in Selangor, Sharjah. This is a small center in Benin, Nigeria. This is a center in Changsha, Polycarpation Professor Taurman Belt and Road Traditional Medicine Center. And of course, there are many mirror centers in China. This is some of the facilities which Professor Taurman has discussed: X-ray diffraction, mass spectrometer, many imaging devices of different kinds. These are some of the animal facilities, imaging facilities, computation libraries, and others. I would like to recognize that we were able to do that through international collaboration. So we have Professor Berzell sitting here, who played an extremely important role in our capacity building in the field of structural chemistry. Thank you very much, Professor Berzell. And what we were able to do is to develop linkages for the benefit of common mission. We can learn from you, and if we can contribute in your learning, we'll be very happy. Thank you very much.
Thank you for marking us on what you have done, a marvelous job, both as a young scientist and a mid-age scientist. You will not say I'm not scientist.
Thank you very much. Thank you.
We will move on to the third because we have the time is limited. Our next speaker is Professor Dr. I will say it in Arabic. That's right. Yes. Well, Dr. Rafat is not, he's from Karachi, a product of Karachi, right?
Yes.
And now he is in Virginia. He's full professor in the Virginia, and his work is on area of cancer, inflammation, cardiovascular disease, and his research emphasis on the central modulation of chronic disease by combination with nutrients and drugs. And he is expert in nutrition, and therefore we'll hear from him. And since I am a diabetic, since we will have diet, but this is diet and cancer, but it's not called diabetes. So you will talk us about diet and cancer gene regulation. Please, Professor Rafat, you have 30 according to my here, the time is 30 minutes. But if you can make it 25 minutes, because we have an online lecture.
All right, well, thank you very much, Dr. Salim Sabri, for the kind introduction. Well, assalamu alaikum and almost good afternoon. I'd like to actually thank the organizers of from University of Jordan for the invitation, especially Dr. Amal Al-Aboudi for a kind invitation, and also like to thank the organizer from COMSTECH, especially my dear friend and brother, Dr. Iqbal Chaudhary, for including me in this program, as well as our science leader, a very visionary leader, Dr. Professor Tayyib Rahman. So thank you for including me in this program. So my topic after these two early morning high profile speeches, I hope this talk, is not that much boring for you. So I'm going to talk about, how diet actually induces certain molecular pathways for cancer prevention. So as Hippocrates says early on, is that let food be their medicine and medicine be their food. And there's also an Indian proverb saying that when diet is wrong, medicine is of no use. And when diet is correct, medicine is of no need. So basically, diet plays a very significant role in health and diseases, right? If you see the cancer, cancer is only like, about 10 to, well, I would say 5 to 10% hereditary. About 95%, 90 to 95% is basically based on non-heritable factors. Profile. So it's okay. It's okay. Okay, and then among those, that was the point of it. So the thing, if you can see that about 70% of the cancer can be prevented easily by like diet, about 30%. 35% cancer can be prevented by taking appropriate diets or by quitting tobacco use. So they play a very significant role in cancer, in various cancer. So cancer is actually, it does not start all of a sudden. It takes a long time. It's a multi-stage process. What happens that you get a mutation, initial a mutation, and then tumor started growing. It's grow very slowly, and sometimes it stops. It stays as a benign tumor until it is another some kind of event, so another mutation. So first mutation, and then you have second mutation. Then it speed ups and grow, and then eventually it leads to a full-grown tumor if it's not treated. And here's just example. How long it takes for different tumors to grow. So as you can see that, like in case of colon cancer, it's like initial, from 5 to 20 years, it takes just for the initiation of the tumor to having like a pre-cancerous stage. Same with other like head cancer, cervix, lung, breast, and prostate, it all takes a long time. So the cancer grows, basically you get like, initiation of tumor, and then it's like mild, moderate, severe, and then carcinoma in situ, and eventually full-blown cancer, where the cancer actually invades through the basement membrane, goes into the blood, and is spread all over the body. So the thing is that all these stages, cancer can stop in any stage. It doesn't have to really go to full-blown if it can be, if the step is taking place, like, if you have a proper diet or something, you can actually slow down and stop at certain stage. So the most significant role in the development of cancer are played by two or a group of genes, which is divided into two groups, like oncogenes or tumor suppressor gene, or we call anti-oncogenes. So oncogenes they are stimulatory for growth and cause of cancer and when they are hyperactive. These genes are basically, they are dominant. So if anything happens to their genes, if there's a mutation and they're hyperactive, it is going to initiate the tumor. Whereas in case of anti-oncogenes, their function is to inhibit cell growth and cause cancer when, so basically they stop cancer growth. And they are actually, their mutation are recessive. As we know, we have two, we have a pair of genes. So if anything happens to one copy, So still the other functional copy will play its role. So in order to knock down the tumor suppressor gene, so this is like a God gave us, Allah Ta'ala, gave us a protective mechanism that we have a mutation, one copy, then we still have the second copy in function. So you have to actually have to destroy both copies in order to destroy the tumor suppressor genes. So what happens that the house cell respond. So basically we have like growth factors, they bind to receptors, and they induce some kind of signaling mechanism. So there's small molecule, they are generated, which we call they are the mediators, and then eventually they cause the changes in gene. So all this information from outside goes into inside, and all these molecules, different molecules, they have some role in the development of cancer. For example, what happens that in certain cancer, there are gene mutation, and we get excessive production of like growth factors. So there are a lot of growth factors. So it means the information is going to go too much, and then the cell is going to be very active. We have cases where the oncogenes or genes, they are expressing a lot more receptors. So the growth factors are not, still the normal, but because we have a lot more receptors, so message is actually increased several times. And then we have things like, molecule like signal transduction, which is like, increases the signal intensity is increased. Or we have like oncogenes or genes which involve in cancer. They also they have transcriptional factors. So they are like the message is actually increased in the nucleus. So we have more rapid transcription of those genes. So all these genes play a very significant role. On the other hand, we have tumor suppressor gene, which actually inhibits the tumor growth. And among them, there are two significant like retinoblastoma protein and p53 proteins. They are highly involved in suppressing the tumor. So I'm going to switch now and talk about some molecular pathways to give you some idea. The dietary agent can suppress the transformation or the hyperproliferation, invasion, angiogenesis, and metastasis of various tumors. So diet actually is a very significant factor. It can inhibit tumor growth in any of these stages. The precise molecular mechanism is still some of the diets, we know how they work, but still we know that they have some effects, they have anti-cancer effect, but we're not sure about the molecular mechanism. And the data from published literature do indicate that diet has a chemopreventive effect. Some of the common dietary compounds which anti-cancer activity is from this list, like curcumin, which is present in what we call haldi or turmeric, resveratrol in grapes, like genistein in soybean products, silymarin is milk thistle, and we have compounds from like allium, like diaryl sulfide or acetylcysteine, from garlic, tomato, red chilies, fenugreek, ginger, and you name it, like pomegranate, apples. All these compounds, every compound has certain polyphenols which has anti-cancer activity, and we will go some of these things. So it's just to indicate all these wonderful things which are present in fruits and vegetables, which can inhibit the cancer. So the way the diet works is they regulate certain intracellular signaling mechanism, and some of these mechanisms are such as, and we go through some of these, like activation of nuclear factor kappa B, or activator proteins things like serine threonine protein kinase B or PI3 kinase and AKT pathway, STAT protein, EGFR, cyclooxygenase. So these are the intracellular mediators which are regulated by diet. And how they are regulated, I'll give you some few examples. For example, NF-kappa B. The way NF-kappa B works is that we have NF-kappa B, which is actually present in the cytoplasm, and it has like 3 units. So basically, NF-kappa B has like 2 subunits, but it is blocked. The activity of NF-kappa is blocked by this I-kappa B. So what happens when the signal comes, when there is excessive production of growth hormones, or there's a kind of a signal which is going to lead to cancer formation or inflammation, what happens? The signal actually phosphorylate this protein here, I-kappa B, and this NF-kappa B is like free. When it is free, is active, it goes to the nucleus. It binds directly to the transcription factor here and induce certain genes. And these genes are involved in all kind of things, which is mostly linked to cancer, such as this causes immortality to the cell. Cell stay longer, it grows longer. Inflammation, angiogenesis process, where the blood vessels are drawn towards tumor. Anti-apoptotic process, the cell die, so it inhibits the process of apoptosis, increases proliferation and tumor promotion. So NF-kappa B is a very central molecule in the cancer growth. So things like curcumin, which is present in turmeric, genistein, in soybean, capsaicin, in like red chilies, resveratrol, grapes, or green tea polyphenol, they all have been shown and proven a very strong inhibitor of NF-kappa B. So they can, I just gave example of curcumin, which can inhibit NF-kappa B. So it inhibits here, it cannot go into the nucleus and actually stop the genes which are involved in the cancer production. So another molecule is AP1. AP1 is also a transcription factor, similar to NF-kappa B, is also present in the cytoplasm as an inactive complex because it's bound to this compound which is shown here in blue. So this is also protein which keep it inactive. But when there's a growth signal, like for forbol ester is a very strong tumor promoting agent. So what it does is it induces activity of various kinases which phosphorylate this subunit, and once it's phosphorylated, it dissociate. Once it's dissociate, it is active. Now it's again the same thing, is active, is translocated into the nucleus, directly bind to the responding element, and then just transcribe some genes. All of these genes lead to the cancer formation. So there are diets, and here's some example which a certain kind of cancer which involve specifically activation of AP1 molecule, and things like, again, curcumin, caffeic acid from coffee, and resveratrol directly inhibit the AP1 binding to DNA. So that's one of the mechanisms the diet can prevent actually activation of this signaling molecule. And the common molecule is AKT, or also known as protein kinase B. What it does is that, again, the signal comes in, signal activate a protein which we call PI3 kinase. PI3 kinase make this lipid here. And once this lipid PI3 is generated, it recruits this AKT to the membrane, and then it get, this PI3 phosphorylate this enzyme AKT. So once it's phosphorylate, it again induce a signal and lead to the growth proliferation. So AKT and PI3 kinase has been involved in a number of cancers. And here, just to show you the list that, so many companies actually are working to make inhibitors of this pathway for PI3 kinase and AKT inhibitors, and all these compounds, there are certain kind of drugs which are made by certain pharmaceutical companies. And again, we have like, curcumin, ECGC, which is present in green tea or other green tea polyphenols. They have been shown to inhibit this PI3 kinase and AKT pathway. So here, like ECGC inhibiting the activation of AKT, it can also inhibit the downstream process of AKT activation, mTOR activation. It can also actually activate P10, which inhibits the PI3 kinase activation. So some of these factors in diet can inhibit this pathway too. Excuse me. And then we have things like MAP kinase. MAP kinase is another signaling molecule. And what happens that MAP kinase also get activated through a series of reactions. So when growth factor bind to the receptor, it activates certain signaling molecule, which activate this MAP kinase, which activate MAP kinase, is actually MAP kinase, MAP kinase, and this is MAP kinase. So through this cascade, once the MAP kinase is activated, it again goes to the nucleus, binding to the responding element, and then induce the transcription of genes which lead to the cancer formation. And then again, this is also one of the area where a lot of inhibitors are certain, these pharmaceutical companies, they synthesize or design various chemicals to inhibit the MAP kinase activity. And again, the curcumin, indole-3, carbinol, which is present in like cabbage, cruciferous vegetables, resveratrol, and ECGC has been shown to inhibit MAP kinase phosphorylation. Another molecule we have is like a STAT protein. Again, the same mechanism that they can activate it, translocate into the nucleus. binding to the responding elements and transcribe the genes involved in cell survival. Again, we have things like curcumin, resveratrol, betaine, which is present in cashew nuts, and capsaicin from red chilies. They inhibit activation of STAT3 signaling molecule. Certain growth factors, their secretion can also be inhibited by things like resveratrol, green tea, polyphenols, and like inhibit IGF-1 production. VEGF production can be inhibited by green tea polyphenols, and EGF production by curcumin, genistein, resveratrol, and catechins. So all these growth factors, they are highly involved in, again, the induction of cancer. So another process in cancer is the metastasis, which is a big problem. And metastasis is mediated through a group of proteins, which we call chemokines. So these chemokines, they bind to this receptor CXCR4. So this is the receptor. So these molecules, these are kind of growth factors. They bind to this specific receptor and induce the metastasis. So the metastasis, basically again, the spread of cancer from from primary site to all over the body. Like you can see there's some signaling pathways, and I think I apologize if you cannot read because their letters are too small. So all this signaling molecule, they induce the chemotaxis, which is a process where the cell migrate towards its receptor, which is expressed on different organs. So like curcumin also inhibit the CXCL, this is a ligand, which induce the chemotaxis involved in invasion and migration. Also reduces the receptor expression, same as 0-1, which is present, I think, in ginger, also inhibit both the ligand and the receptor expression, so that they are involved in the inhibition of metastasis. Other pathway are involving the cyclooxygenase 2, which is basically uses the arachidonic acid in cell. So this cyclooxygenase 2 generates mediators, which are inflammatory mediators. So these are the prostaglandins, prostacyclin, thromboxane. These are the inflammatory mediators, which eventually, if it's not inhibited, they lead to cancer formation. So the COX-2, the number of attempts has been made to inhibit the COX-2 activity, and we have diets, things like resveratrol and nitrosomethylbenzylamine and ECGC. They are shown to inhibit the COX-2 activity. So again, they can inhibit the inflammation. The process of angiogenesis is a process where the blood vessels actually are formed to supply the nutrients to the newly developed tumor. So this tumor, this is a primary site. Tumor goes through the basement membrane, goes into the blood, localized to the secondary site. And for the survival of this tumor, it draw the blood vessels. And for drawing this blood vessel is the vascular growth factor, VGEF, plays a very significant role. So we have, again, the dietary components like curcumin, ECGC, catechin, and resveratrol. They process the inhibition of this angiogenesis by inhibiting the VEGF signaling process. Cell cycle, again, we know that in cancer, the cell replicate, multiple times. So what happens that, with cell, during cell cycle, cell goes from G1, GS, G2, and M phases, and all these phases are regulated by this tumor suppressor protein, retinoblastoma protein. And it's actually in the normal cell, this cell cycle is inhibited. But once this protein is phosphorylated, then cycle gets continued and goes on and on until the signal comes to a stop it. And we have things like, curcumin, resveratrol, diisogenin, thymoquinone, genistein, ECGC, and GSC. This is grape seed extract. They also shown to inhibit the mediators of cell cycle regulation. So that can be inhibited by using proper diets. Apoptosis is a process where a normal cell actually get a signal, and what we call is like a programmed cell there. So any like anti-cancer drug we take, what they do is send the signal to the normal cell, and the cell is started actually dying. So it disintegrates and dies and undergo lysis and disappears. So apoptosis is also a very regulated process. What happens, the number of caspases, they get activated, and these caspases lead to the disintegration of cell. And we have dietary components. And again, I apologize if you cannot read, but just to summarize that, like curcumin, we have resveratrol, ECGC, these has been shown to induce a mechanism which actually can lead to apoptosis, or they can induce the mechanism which actually inhibit the process which inhibit the apoptosis. So in both ways, it's the cells dies. So these are certain genes, but in addition to gene mutation and all that, their diet also plays a significant role for regulating the epigenetics. So epigenetics is a process where there's no change in gene, but there are processes above the gene which are modified. What we call is like histone modification, DNA methylation, and the release of microRNA. So these are involved in gene regulation. So what happens that we know that histone get acetylated. When histone get acetylated, the chromatin opens, and then genes start transcribing. So this involved in gene transcription. Whereas DNA methylation is a process when the DNA methylated and actually blocks the gene expression. So some of these tumor suppressor genes, they are methylated, so they do not function during cancer. Whereas the genes, oncogenes, they are acetylated, so they are expressed more during cancer. So here is just to show you what happens during DNA methylation, that the DNA methyl transferases actually transfer a methyl group to cytosine. So cytosine is get methylated. So in normal cell, what happens that there's some genes like oncogenes are inactive because they are methylated, but the tumor suppressor genes are non-methylated, so they are active. But that get reversed in cancer. In cancer, what happens that the oncogenes, they are demethylated, so they become like so they are sorry inhibited, and sorry, they are active. So they are demethylated, they are active, whereas the tumor suppressor gene, they are methylated, so they become inactive. So the process gets reversed. And in histone modification, we know that if you put a SL group to histone, then because of the SL group has a negative charge, so negative charge repels, and then you get the chromatin opens, and you get the gene transcription, and most of the genes are the oncogene will lead to the cancer formation. Whereas histone deacetylases remove the acetyl group, and then chromatins get compact. So this is the like an entire oncogenesis process which involves in like inhibition of in the process of like activation of like tumor suppressor genes. So there are things like in diet which have both induced DNA methylation or histone modification, or there are certain dietary components which actually do both. Like they are also good DNA, inhibit the DNA methylation, well as also inhibit the histone modification. So again, these compounds, the dietary compounds, they are either inhibitors of DNA methyltransferases or histone acetyltransferases, or they are the inhibitor of histone deacetylases, and all these processes actually leads to inhibition of cancer formation. So one of the examples is like ECGC. How it works is that, as I said, that DNA get methylated by the DNA methyltransferases on cysteine. So what ECGC does is that this is like, S-adenosyl methionine is serve as a methyl donor. So methyl group comes from this S-adenosyl methionine, and it becomes converted to S-adenyl homocysteine. And during this methylation, we get effects on DNA. But what happens that the ECGC is a cortical amine. So this is cortical amine methyltransferases. This actually uses S-adenomethionine and methylate the cortical amine. So we can produce this S-adenyl homocysteine, and which is also inhibitor of DNA methyltransferases. So there are two things. One is that actually this S-adenosyl methionine is taken away from this methylation process, and again the generation of this SA is also inhibiting this process. So ECGC, the green tea polyphenols are very potent DNA modifiers. So SA is a potent inhibitor of DNA methyltransferase, as well as ECGC directly inhibit DNA methyltransferase. And we have also when we have the hypomethylation of tumor suppressor gene, then we get the expression of certain genes, and which are kind of re-expressed. Same as we have like microRNA. So microRNA, these are small RNA, and what they do, they are like inhibit the tumor suppressor, so they are like oncoRNA, or they are actually inhibit the oncogenes, so they are anti-RNA, and they are involved in regulating all different organs. So again, we have a number of dietary compounds, and again, it's probably difficult to read, but just to make this story short, that we have a number of dietary components which directly modify the epigenetics. So they're not actually doing anything to DNA structure, but they are inhibiting the processes which involve the gene expression. So again, things like curcumin, ECGC, they has been shown to actually regulate the release of certain miRNA in certain cancer, which lead to the kind of regulating certain cancer. So I just want to, I don't know how much time I have. I'm not sure.
2 minutes.
2 minutes. Okay. So this is just an example that, there are a number of dietary components which can induce the miRNA, which are involved in inhibiting the cancer process. So again, just a few things like, American Institute of Cancer Research, they have certain recommendations. Their recommendations are that choose a diet which is rich in variety of plant-based foods, eat plenty of vegetables and fruit, maintain a healthy weight, and be physically active. Drink alcohol only in moderation. I'm sure there's not the problem here. Select food low in fat and salt, and prepare and store food safely. The American Cancer Society has similar kind of guidelines, is that choose food that help maintain a healthy weight. When you eat away from home, choose food low in fat, calories, and sugar, and avoid large portions of food. Eat a smaller portion of high caloric food, but be aware that low fat or fat-free does not mean they are low calories. That low fat cakes, cookies, and similar food are often high in calories. Substitute vegetable, fruits, and other low-calorie food for caloric-dense food, such as like French fries, cheeseburger, pizza, ice cream, donuts, and other sweets. Instead of eating that, you can have some kind of fruits and vegetable. Eat a healthy, healthful food with an emphasis on plant sources. Eat 5 or more serving of a variety of vegetables and fruit each day. Include vegetables and fruit at every meal, and for a snack. Eat a variety of vegetables and fruit. Again, limit French fries, snack chips, and other fried vegetables products. Choose 100% juice if you drink fruit or vegetable juice. So these are some of the recommendations. Some of the lifestyle changes, especially for breast cancer, like reducing alcohol consumption. Breastfeeding actually is a thing which can modulate cancer progression. Avoid obesity. Obesity has a very strong link to breast cancer. Eat more vegetable and fruit. Limit intake of red meats and avoid obesity. The reason I put it that in Jordanian, I found that the breast cancer is too high in the Georgian population compared to other cancer. So what it is, it's simply eat less crap, which is like avoid carbohydrate, refined sugar, artificial sweeteners, and processed food. Eat food which is fruits and vegetable, organics, lean protein, omega-3 fatty acid, and drink water. So this is just to show that there's a fruit or food for every organ in the body. Allah subhanahu wa ta'ala gave us fruits and vegetables to basically maintain the health of every organ in our system. So I stop here. Thank you very much for listening, and then I'd be glad to answer any question you have. Thank you.
Thank you. Thank you.
So I hope I finished it on
I
time.
think everyone of us now prepares to watch his diet very well.
Okay, thank you. All right, thank you.
Let's thank our speakers, Professor Atta, Professor Rafa, and we end here the session. We will have 20 minutes, but now we will have 10 minutes only. So that will come back after 10 minutes because we have 2 lectures online from China. And therefore, please be on top of that. Thank you very much.
We need to
introduction.
Okay, Auzubillahi minash shaitanir rajeem, Bismillahirrahmanirrahim, Assalamu alaikum, and good afternoon, ladies and gentlemen. Let me welcome you to this free lunch session. And in this session, we have two very distinguished speakers from China. Both of them have made tremendous contributions in the field of spectroscopy, natural product chemistry, and drug discovery and development. And first of them is a very dear friend of ours, Professor Dr. Wai Weng. And Professor Dr. Wai Weng is known to us since last 1 decade. I hope Professor Wai Weng is online. Professor Wai, are you online, brother?
Yes, Professor Dr. Iqbal.
Thank you very much. Thank you for accepting our invitation. And let me just very briefly introduce you to Professor Wong. He is a Yangtze River scholar from this distinguished professor in Hunan province. He is the doctoral supervisor, director of traditional Chinese medicine and ethnomedicinal innovation and development international laboratory. He is the dean of School of Pharmacy, Hunan University of Chinese Medicine in Changsha. He is also the visiting professor at our center in Karachi. Also, he's a visiting faculty at Shanxi University of Chinese Medicine, Southwest Minzo University, consultant of Luzhou Municipal Government. He and his group work on the research of material basis of TCM in Hunan province and new methods of screening the biological activity of traditional Chinese medicine based on specific drug targets and research on the targeted synergistic effect of many bioactive constituents of TCM plants. Dr. Wong is a principal investigator the principal investigator of 30 national, provincial, and international cooperation scientific research project. He has developed more than 100 standards about national industry and other TCM enterprises for TCM enterprises. He successfully published over 300 research publications, obtained 13 patents, and won 7 provincial scientific excellence awards. He serves as an executive editor of Current Traditional Medicine. He's the chief editor of Current Chinese Science Natural Product Section and associate editor of Helion, which is another very prestigious journal in the field of biochemistry, molecular biology, and cell biology. More than that, Professor Wong is truly an international citizen because he believes on international values of science. His group is very diverse and he has students from Bangladesh to Nepal to Africa and different countries. This is my honor to introduce my dear brother and friend, Professor Wai Weng from Hunan University of Chinese Medicine. Professor Wai Weng.
Okay, thanks, Professor Iqbal, kind introduction. We would like to thank Xin committee for inviting Professor Dr. Wang to take part in this conference. Dr. Wang feels so sorry that he can't report by himself because he is busy with other things. So today I will report on behalf of him. I'm Dr. Yu Pei Yang as assistant with Dr. Wang from Hunan University of Chinese Medicine. I'm honored to be here, has a chance to share our research group work. The title of my presentation is Natural Products Research on Hunan Ethno Medicine. Firstly, I want to share some beautiful landscape photos to all of you. The beautiful Zhangjiajie, Huping Mountain in Hunan province are both located in Wuling Mountain area, which is important treasure house of plant resource at 30° north latitude. The unique geography environment and climate bring rich and colorful medicinal plant resources, which produce complex and diverse chemical components. And these chemical components are the base of pharmaceutical substance, Hunan as an ethnic medicine for disease prevention and treatment. Therefore, our research story begins with the colorful ethnic medicine. Here are the pictures of Hunan ethno medicines that our research group has studied and is currently studying, like 黑老虎, 白山崎血统, and so on. And now our research group has identified 1,000 compounds, including more than 20 new compounds. There are the structures shown in here. Our research status is tracking in Hunan Tujia as medicine literatures and digging the efficiency, focusing on xue and qi herbs, and making efficiency research on substance or breakthrough for stringency of modern research on ethno medicine. There are many aspects to about my presentation, starting from basic research on pharmaceutical substance of TCM and ethno medicine. First is first of all, I want to introduce the basic research of substance of Tujia xue herbs, which has the effects of expanding winding and activating blood, enemy stasis and swimming, stopping bleeding and relieving pain, such as 一口血, 血蜈蚣 and 血桐. Why we call the Tujia xue herbs named blood herb? Because we can find these herbs whose surface and section were red and brown, taken an analog. So we call the xue herbs called blood herbs. The first xue herbs is 血桐, which is originated from the family Stringiaceae. 血桐is a rich source of lignans and tritopenoids. In Wuling mountain areas, the stems of 血桐 was used to treat eye, rheumatoid arthritis, and so on. There are highly oxidated nanostin tritopenoid and 3 for recycle cycloaltone tritopenoids isolated from 血桐. There are single crystal structures shown in here. And 6 new tritopenoids, we named the 血桐nectin A to F, have been identified, which structures determined by 1D and 2D NMR spectra. Cytotoxicity activity screening showed that 5 compounds showed struggle toxicity to cancer cells with the IC views less than 10 micromole. And 6 new strain tritopenoids named 血桐 dinectin A to F. There are another 6 new tritopenoids named 黑老虎, named 血桐素 A to F. Except tritopenoids, we also have been identified some lignans and cis-tritopenoids. Additionally, 51 compounds have been isolated from the leaves of 血桐, including 8 new compounds. On the basis of pharmacochemical research, our research group conducted the quality control of 血统 and established the content determination method of 6 main compounds. At the same time, the isolated compounds will also screen some activities like anti-RA, anti-tumor, antioxidant, and antiviral. In the anti-RA results, we can find 血统素 showed significant activity with the IC values of 9.12 micromole. And the 6 compounds showed significant anti-tumor activities. 8 compounds showed anti-inflammatory, antioxidant, and antivirus effects. 血统 is one of our typical 2-家 herbs. It is mainly used in treatment of RA. The anti-inflammatory and anti-genic effects of 血统 acerola extracts were studied. The results showed 血统 acerola extract with 50 to 400 milligram each kilogram doses showed excellent anti-inflammatory and analgesic effects. The analgesic effects of 血统素 with 39 milligram each kilogram equal to morphine with 7.8 milligram each kilogram. 血统 acerola extract also has the neuroprotective effects by separate of oxidative stress, inflammation, and apoptosis. Our group also systematically evaluate the anti-RA efficiency of 血统 acerola extracts, and the results show that 血统 acerola extracts with 200 and 800 milligram each kilogram dosages show excellent anti-RA effects, which can inhibit the bone erosion of ulcerated joints and protect the joints against cartilage destruction and bone erosion in AIA rat model. 血统素 is a typical tritenoid which isolated from the 血统, and also it is the highest constituent in 血统. The way our research group also reported the X-ray structures for the first time. Anti-RA effects of 血统素 were studied in vivo and in vitro. The results show that 血统素 can inhibit the souvenir high pressure, bone erosion of ulcerated joints, and protect the joints against cartilage destructions and bone erosion. Also, 血统素 exhibited good biological safety and have no obvious toxicity and side effects on the heart, liver, spleen, lung, and kidney. Absorption, distribution, and metabolism in vivo synthesis of 血统素 were studied. Absolute oral bioavailability of 血统素 was about 79.3%. It is treated by phases with 41.7% and its calculated excretion rate at 48 hours. A total of 15 metabolisms were identified by a 3-step analytical strategies. About 血统, we have published 31 essay papers and obtained 2 patents. Next, I will introduce another 2-家as a medicine. It's a 2-家 qi herb. Which is another important focal medicine, like 白三七, 扣子七, craft, and so on. The first is 白三七. 白三七 is similar to penlagensin, possess enhanced immunity, disappear white, and demomodify effects. It is also be collected in China Pharmacopoeia 2020 at Zhou. We also did the phytochemical investigations on 白三七, and now 45 compounds are being identified, including 8 new compounds. it is recently mentioned that 3 compounds with a unique pattern examining tuning moiety in the new compound. Next is 积血期. 积血期 it has the effect on relieving rigidity of muscles and activity color trators, stretching, blending, and relieving pain. We also did the phytochemical investigations, and now 9 new flavonoids, including 2 carbamazepine 4 conjugate flavonoids, have been identified. And our research group designed a new fluorescent enhanced nanoprobe for the extreme screening of the compounds. The results showed a compound G5 that can induce the apoptosis of colon cancer cells by inducing the release of a large amount of cytochrome C. The rules of casual carcinoma in China we call 黑老虎. As a tool as a medicine, it also be used to treat RA. And now we did the phytochemical investigations about 黑老虎roots and fruits. So far, 172 compounds have been identified, including 45 new compounds. And there are 3 new ligands. We named 黑老虎ligand A to C, which possess dibenzoyl skeletons. There are 5 new ligands named 黑老虎素 A to E. We did the hepatoprotective activities assay of 68 ligands that are isolated by our group. According to the assay results, we did the SAR of dibenzoyl ligands against the APAP-induced liver injury in hepatocyte cells were analyzed. At the same time, we found a new dibenzoyl ligand 黑老虎素磷 B can animate APAP-induced hepatotoxicity with oxidative stress inhibition and activating the NF2 pathway in vitro. And one dibenzoyl ligand ISBA can animate cardiac ischemia reperfusion injury through activating GR-dependent risk signaling pathway. And one new dibenzoyl ligand 黑老虎素 C that can prevent the development of BGC to 3 cells in vitro and promote cell apoptosis by P53 and mitochondrial apoptotic pathway. Except the ligands, we also isolated 32 tritopanoids, including 10 new ones. Also, we found 1 new tritopanoid named 黑老虎acid G that can inhibit the proliferation, introduce apoptosis, and animate inflammation in RAF cells with separated NF-kappa B pathway. Additionally, we obtained 20 cis-quintonoid compounds, including 2 new ones. One cis-quintonoid can separate inflammation by inhibiting the NF-kappa B and JAK2 STAR3 pathways. And also, 65 compounds have been isolated from the fruits of 黑老虎, including 20 new ligands. And next is about cytotoxics about the compound isolated from 黑老虎. The results showed 6 tritopanoids exhibited significant activities against the 5 different human cancer cell lines. A series of novel molecular probes based on specific targets were designed in combination with fluorescent probe technique to screen activity compounds related to inflammation and cancer. The results show 6 compounds had been screened that with a significant activity. Next, while focusing on the study to medicine, We also carry out the study on Dong medicine, which is also belonging to Hunan medicine. 清钱柳 as a Dong medicine called morning tree or sweet tea tree. It can be used to treat hypoglycemia and diabetes mellitus. And now 22 new compounds and 28 new anoles were isolated from the leaves of 清钱柳. Here are the 8 new compound structures. Here are another 40 new tritenoids. We named the 清钱柳素 A to M. A series of enzyme activators related to tumor therapy were screened from 清钱柳using a novel molecular probe technique. Next, we did the research on pharmacodynamical substance of TCM. The first is huangjin, which has the effects on supplementing qi and nourishing yin, notifying kidney and spleen, nourishing the lung. And now 92 compounds are being isolated. Among them, we found 6 homoisoflomoalkanoids with GABA structures that we found for the first time. The second is 金丝黄菊, which is a food and drug uses. And now 111 compounds have been isolated, including 29 new compounds. The second part is about research on new methods for screening activity of TCM based on specific targets. And now our group has established the plan for rapid separation and identification of TCM active ingredients. Also established the plan for screening activate of TCM based on specific targets. And a variety of novel molecular probes were designed to explore the diverse and specificity of TCM and asterose drugs. Here are one case. An NIR probe have been designed, has been developed to detect liver bacteria and injurious with specificity, high sensitivity, and visualization. The probe was successful, used to potent and safe production agent to efficiently treat drug-induced enteropathy in vivo. The third part is research on target synergies of active ingredients in TCM. Biomaterials are used to modify the structural surface of active natural small molecular compounds in traditional Chinese medicine and ethnic medicine, so as to achieve precise target intervention treatment. And the first one case is based on 血统素 as an active substance. The nano drug delivery system was contrasted to enhance the targeting ability to 血统素 in the AIA rat model, which can improve the bioavailability, reduce the toxicity, and clarify its anti-RA inflammatory mechanism. Thus laying a foundation for the research and development of a safe and effective anti-RA national innovation agents. The second is targeted and enhanced therapy of anti-tumor components from Baisanqi. The first is hybrid cell membrane coating or nano complex loading a compound. The compound isolated from Baisanqi for a combinational therapy against the breast cancer system with CE6. The second is hybrid cell membrane nanoparticles noted compound. The compound also isolated from bisanchi for breast cancer combination therapy. The first part is about research on quality standards of traditional Chinese medicine, improve the quality standards of Chinese medicinal materials, and the Chinese patent drug and support 6 companies to upgrade their product quality. Here one case, quality assessment of and Q-mark discovery of Juhua were achieved by combined chromatograph effect relationship and bioinformatic analysis for the first time. This approach is powerful for describing the flaws and for providing clinical guidance. The fifth part is about international communications. Since 2013, our research group has been activated serving the National One Belt, One Road initiative, and also carry out multi-dimensional cooperation with Professor Dr. Art and Professor Dr. Ikubo research group. Certain achievements have been made in building an international cooperation plan for constructions and also training of international high-end talents. Finally, it is a great pleasure to invite you to the 6th Hunan International Conference for Innovation and Development of Biomedicine and Chinese Medicine, which will be held from November 1st day to 5th in China, Hunan Province, Changsha City. Since 2015, we have successfully held 5 international conferences on history. So we are truly glad to welcome all of you to China, to our beautiful university. So that's all. That's the end of my report. Thanks for your listening.
Thank you very much. Thank you very much, Professor U.P. Yang, for presenting excellent work of your research led by Professor Wei. And I know that the excellence of work being conducted in your laboratory is now internationally recognized, and you've been publishing in very high impact factor journal. I think we have some time. If you have any question to Professor Yang. Apparently not. Professor Yang, kindly convey our best wishes to Professor Wong, and we are looking forward to seeing you in Karachi. There was a visit planned. Hopefully this visit will be planned soon, because I believe that there is a delay in it. Yeah.
Okay, thanks, Professor Ikubo.
Thank you very much. Thank you. Okay, so my dear brother Jalal has assigned me to introduce the next speaker also, and who is certainly one of the finest chemists who has used metabolomics based on NMR and mass spec to the best of its application one can think of. Professor Kai-Feng Hu, who is known to us since over a decade, and we have had the honor of knowing him, not only because of his contributions, but also his very friendly attitude. Professor Hu is the professor of bioorganic chemistry. He is expert in NMR spectroscopy as well as metabolomics by using different methods. His introduction, briefly, I would say that he's the principal investigator of Chengdu University of Traditional Medicine. He has recently moved from Kunming to Chengdu, China. He received his PhD degree in 2004 in solution NMR methodology from Swiss Federal Institute of Technology in Zurich, Switzerland. In 2005, he joined National Institute of Health, USA, for his postdoc in structural biology and proton NMR spectroscopy. During 2009 to 2012, he worked as an assistant scientist at MRFM, University of Wisconsin, where he began to focus on method development for the metabolomic study by integrating NMR and LC-MS. In 2012, he was appointed as a professor, principal investigator at Kunming Institute of Botany, Chinese Academy of Sciences, and established his independent research group. Dr. Hu has very strong interest in NMR theory, hardware, and applications. His expertise in the field of research and interests include metabolomics, structural biology, natural product chemistry, and bioactive compound of TCM plants. He also worked on method development for quantitative and qualitative analysis of complex mixtures, largely plant mixtures, primarily using NMR spectroscopy and LC-MS, together with other biophysical and analytical methods. Professor Hu, I appreciate your time for this event. We wanted you to be here in person, but we know that you're extremely busy, and I request you to kindly present your lecture. Thank you very much.
Hi, everyone. Thank you, Professor Ikuba. Thank you for the nice introduction. So can you see my screen?
Yes, yes,
Okay.
we can see your screen.
Okay, so I will go ahead. Okay. I'm very glad to have opportunity to attend this meeting. So today I'm going to talk about 2D NMR-based metabolomics. I will introduce the general workflow of how to do 2D NMR-based metabolomics. So as you know, for metabolomic study, our goal is to discover the biomarker. But what I want to emphasize here is to discover the genuine biomarkers from metabolome. So there's a long way for you to travel from what you get is what is exactly in the metabolome, or in some case, maybe what you get is not what is the true thing, the background truth. So I show you the journey, the pathway you have to travel, how to, from the metabolome, you discover the biomarkers. So from the metabolome, you have to prepare your samples. Once you get the sample, you have to do data collection. Depends on what kind of platforms you have. You can collect LC-MS data or NMR data, and you do the so-called data pre-processing, and you do some analysis, multivariate analysis, or univariate analysis, and then you have make some rules to select differential variables, and then you try to identify, or I call it the qualitative interpretation, or structure isolation, or identification of what you get. So in such case, you find the biomarker, but you have to make sure what you find, you discovered, is rarely seen in the metabolome. So you have to do the biological verification, because here, whatever you get from the data, from the mathematical work, is just the statistically. So it shows statistically significance, but it doesn't mean it is a causal relation. So from this long pathway, for any of this step, if you lose the biological information, so your result, or the result you obtained, could be wrong, or is irrelevant to what is the truth, the background truth. So I'll make it short. In brief, so the metabolomics study is try to find what have changed for the compared patterns. So in this case, you have to consider the thing in 3 layers. Why is called omics? So which means your result has to be statistically significant. And what changes? So you have to identify, you have to make sure you can elucidate or the chemical identity of the metabolites correctly and changed, which means you have to be quantitatively accurate. If the quantification is somehow not accurate, or in that case, the conclusion you draw could be also wrong. So I make a kind of like a small short summary like this. So what contents change, which in 3 different sides, which means you have to do it quantitatively correct. You have to determine the chemical ID, and the concentration or the contents change means is quantitatively has to be correct also, and the change has to be statistically significant. So in this way, I want to show you a comparison of the normal 1D proton NMR and the 2D HSQC. So for very complex samples like the metabolism or the effects of Chinese traditional Chinese medicine, there's a severe signal overlap. So when you use the 1D proton spectrum to do the metabolomic analysis, one thing, first of all, you can see you cannot make sure your quantitative information or is correct, or because the baseline, or even you integrate some signals, it could because of the overlap. So your quantitative information could be not that accurate. So there's a brief like a comparison to show the difference of 1D and 2D HSQC. So for some signals, like you look at the 1D, it seems like a rare 1D single peak, but actually we do this 2D to expand this along this carbon dimension. You can see actually these peaks, they are not just 2 or 3 peaks. There are multiple metabolites underlying. So in this case, even you analyze the peak volume of this 1D peaks, it cannot represent only one metabolite, because it's a kind of a mixture peak from 2 or 3 metabolites. In such case, will make the metabolomics analysis very challenging or even impossible. So we propose you for such a complicated complex case, it's better to do a 2D NMR based metabolite study. So another case is for the metabolite identification or your corridor quantitative interpolation. Because of the overlay of these peaks, actually the 1D, even you have a good starting case, I give you an example, such like this peak is a kind of a low overlapped signals of 2 compounds A and B, and if you for some case you have a good case, you identify it as the signal from compound A, and you even do the spiking to confirm it. Of course, when you add A to the mixture, your spiking show you the conforming result. But even in such case, doesn't mean your result is correct, because anyway, either you add A or B spiking into the mixture, you always get the signal increase. So even you kind of like you're confirmed by 1D spectrum spiking, is you still can have possibility to get the false positive result. So to avoid all this the confusion during the sample preparation and data collection and the interpretation, we present one concept. So whatever from this metabolism, from the initial sample to the final end of the data, you have to keep the biofidelity. What I mean biofidelity means you have to keep what is the restitution in the metabolism sample, and you have to carry it over to the data and to the data analysis, so on. So the first thing for the sample preparation step, you have to pay attention. For some metabolites, maybe because of the solvent that you choose, maybe it's insoluble of the metabolites. So in such case, if you use some improper solvent to extract your metabolites from the mixture, so in fact, you will lose some of the metabolites in the sample. So later on, whatever, even you use LCMS or NMR, you cannot collect the data information from the specific metabolites. Another thing is, again, because of the volume, let's say if you use too less volume of your solvent, you may have an incomplete extraction. Or you use, in other case, if you use too much of the biomass or biomaterials at the beginning, even you extract all of them, but at the final stage, you have to put all this in dissolved or redissolved in the determined solvent in the NMR tube to be able to mirror the NMR signal. But in this step, if at the beginning you took too much materials, you could oversaturate the stuff you extract in the 500, let's say 500 microliter determined solvent. So in this case, for sample preparation, we propose you have to pay attention to 3 important aspects. Firstly, you have to choose the proper solvent. The second is, even you have the solvent prepared, you have to add enough or proper amount of volume of the solvent for certain amount of the materials. And for each of the NMR sample, let's say, because the final volume is only 500 microliter, you cannot put too much of your metabolome or the materials of traditional Chinese methods. So, which means you cannot oversaturate the final solvent. So I'll give you some details how to make this. Like this is the case, we prepared the sample of cell metabolomes. We first choose 1.5 to 10 with a power of 6 in the cells. And we use 5 different solvents to try to extract them. And for each of this extraction, we collect 1D protein spectrum and make an integration of this 4 region. Basically, we just include the solvent region. So in this case, we compare the integration of this different region. We can see for different solvent, absolutely, it's very obvious. If we use this MWC1, we actually is the methionine, methanol, water, and chloroform mixture at the ratio of 4 to 1:1. So at this ratio, this is the solvent. This is the mixture solvent. We get the maximum extraction of the NMR signal from the same amount of the cells. So we, in such a case, we will choose this solvent as the proper solvent for the extraction for the coming experiments. So the next thing we have to try out is we still use the 2.026 cells, and we try with different amount of the solvent. The solvent we choose from the first stage. We will see if we didn't make complete extraction. Let's say, for example, like use just 400 microliter solvent, which means if we increase the volume of the solvents, let's say to 500, 600, or 800, we could have the possibility to increase the amount of the signal or the materials we finally put into the NMR tube. So in such case, we can see when we have the volume reach to 600, we kind of like reach a maximum. Of course, there's some measurement noise. So in this case, we decide for this amount of cells, we use somehow a little bit more than what we get. We use 700 microliter solvent to make sure we can completely extract all the metabolism from the cells. So the third thing is we simply scale up this thing. We use starting from like 2.0 10 to 6 cells per 700 microliter solvent, and we scale up, which means we use more cells, and also corresponding we use more much bigger volume to extract this. And by the final stage, we dry this and we put all things into 500 microliter the NMR solvent. So if the final stage, the 500 microliter deuterium solvent can only, let's say, dissolve certain amount of cell metabolites, which means the later stage, the overall amount of the metabolites will not increase anymore because it's kind of like a saturate. So in such case, we find a proper amount of the cells. Actually, the cells is proper to prepare the NMR sample, which we choose the kind of like the overall linear stage, because if we later stage, when we do the metabolic study, we want to see the difference in different sample. We have the possibility to see the change or the amount change of different metabolites. If you put too much of cells in the samples, maybe one of them could be saturated. In such case, even the original amount in the cells is different, but you cannot observe different single intensity when you do the measurement. So this is the overall the kind of like a protocol to how to make the cell metabolism sample for NMR study. And these are after these steps, we determined our final protocol. So it's for like 2.0 10 to 6 cells, we use 700 microliter. This mixture solvent, and for each NMR sample, we use like 2.0 10 to 7 cells, and we dissolve in 500 microliter solvent for NMR measurement. So once we determine the NMR sample preparation conditions, we can like look back forward to decide to try how to make the cell culture. So in such case, we know our final goal is to reach a 2.0 10 to 7 cells per NMR sample. So seeing the density of the cell culture at the beginning, we determined is kind of like 5.2 to 10 to 6 cells, and we kind of fix the cell culture protocol for like 24 hours, and you treat by you change the medium and grow another 24 hours. So total is like 40 is 72 hours, and then you reach. We know we tried this. So and you reach 2.0 10 to 7 cells. So at this stage, you prepare the NMR sample for measurement. So and the other small other thing you have to pay attention is we try this how to harvest the cells. Usually you can do the conventional tryptosanization of the cells, and but you can also do the scrapping. In such case, you can avoid the metabolite leakage if you do the tryptosanization of the cells, because the cells is kind of like broken, so you could leak some metabolites. So for this is the sample preparation procedure for the cell metabology, cell metabological study, cellular metabological study. So for the traditional Chinese medicine sample, for the sample preparation of traditional Chinese medicine is pretty similar. We also select or we try different solvent to find the proper solvent. So I will not go into the details how we prepare the prepare the sample of traditional Chinese medicine. So we also choose the proper volume. So we fix the biomass to the solvent ratio, like 1 to 200, the weight to the volume, and also the amount of the materials you use for each of the NMR sample. So at the next stage, once you have the sample ready, you can, again, this is the kind of the picture. You click 1D or 2D. Just now, I already elucidated the advantage of the 2D NMR spectrum. But as we also know, the disadvantage of 2D NMR is you have to spend a longer time to collect the data. So that's why we have to spend pretty much time to optimize the sample preparation to make it as concentrated as much as we can. So in this case, you can make the measurement time to be reasonable. So in this case, for our case, if we collect a 2D proton carbon HSC to get a reasonable signal to noise ratio, and within reasonable time, let's say we try to control it around 2 hours or sometimes 3 hours, we can get the enough signal in 2D HSC for the analysis, for the metabolism analysis. So this is data collection. And now the next step, which is the personally, I think is the really important step, or the very critical step, is the data pre-processing, or we call it deconvolution. What it is actually we did all the same thing for in the structural biology study, like to do the protein spectrum analysis, like we analyze the NOE spectrum for the structure calculation. We have 2 steps. One is the peak picking, and then is the quantification. So for NOE, I think we don't really need that accuracy to do the structure calculation. We in our group, we normally use the Sparky or some other softwares. It's pretty good. You just get the peak position and the very rough volume or the quantification information. But for metabolism study, we have more demanding requirements. So we have to make sure the peak picking, not really make sure, we have to improve our peak picking procedure and the quantification procedure to try to decrease or lower the false positive and increase the accuracy of the quantification. Why? Because all the peaks were used for analyze to get the chemical ID. At the beginning, I emphasized 3 aspects. The chemical ID of the differential metabolites. So if the peaks you pick is not a real thing, is like false positive, you could get the false differential metabolites at the later stage. And also why the accuracy of the quantification is also important, because as we know, if we do the data analysis, the so-called multivariate analysis or univariate analysis, all of this depends on the quantification information of each of the signals or the peaks. So in such case, for metabolism study, both peak picking and the quantification is much more demanding than the what we did in the structural biology study, like you use Sparky for to get the volume to calculate the protein structure. So for the peak picking and the quantification, the most important thing, I think, the first stage, you have to choose the proper model. Of course, you can use the direct integration or summation, but as I said, even we use the 2D HSC, still there's a sphere of life. So the direct summation or integration usually is not that accurate or is not that reliable. So we prefer to use given the peak model to do the so-called deconvolution. But usually we think NMR signal is kind of like have a Lorentzian profile, or you can say it's like a mixture. What I mean mixture here is like weighted average, or somehow is just some weighted average between Lorentzian and the Gaussian. But in fact, you make such a simple peak model, is most time you cannot get pretty good result, which means it's not accurate, it's incorrect. And another thing is because whether it's Lorentzian or Gaussian, we have to do or fitting. Usually it's a nonlinear fitting. So for the nonlinear fitting, you have to guess the initial value of the parameters, like the Lorentzian, you have to guess the relaxation decay rate, or the Gaussian, you have to guess the sigma value. So you use the gradient-based optimization, either the gradient descent, or you calculate the Jacobian matrix to make it gradually move to the rear. You try to find the rear parameters for your peak model. But if you use the incorrect peak model, you cannot, most time you cannot get pretty good results. So by theory, the real peak model of NMR is actually a void peak line shape, which is a convolute Lorentzian and Gaussian. So this, because for the peak model, the void profile in the frequency domain is not a simple function, it's a complex function. So, but in the time domain, you can write this peak model, the void peak, as a simple as a product of the relaxation of the simple relaxation and or Gaussian dispersion. So this the real NMR peak model is a void profile. Here is what I showed is a simple Lorentzian line shape and a Gaussian line shape, which for some cases people use it to do approximately estimate the volume of peak volume. So, and the other case, before some other scientists also proposed some other models, like they called generalized Lorentzian. Actually, even the generalized Lorentzian, they just expand the range of the line shape is they expand the line shape like 3 times wider than the Lorentzian and the Gaussian shapes. But anyway, this is a very rough approximation of the real NMR line shape. I don't think you can get a pretty accurate result. So before, we also tried a hybrid time and frequency domain maximum likelihood reconstruction of like 2D NMR spectrum. So here, like 2 slides ago, I show you the time domain model of Lorentzian and the Gaussian. And in such case, we make it a simple mixture. We can see the relaxation rate, not relaxation, the exponential of the t in the Lorentzian is t, just the minus t, but in the Gaussian is minus t squared. So we mix or kind of like a very simple operation. We make the decay as a function of t raised to eta. Eta is, we call it the relaxation decay power. So in this case, the eta obviously is choose between t1 and t squared. So we fix this eta at by experience at 1.2. But let's say we try to use this in rare case. Still, we get pretty much quite a lot of the false positive. So which means actually this model is still not that really good. So we are still working on this. So next step, once we get the peaks, we have to do the peak matching and the alignment. This is a picture to show the peak matching and alignment. What does it do? So say you have different groups of samples and you collect all this 2D NMR spectrum. And for each of these spectrum, you have a bunch of peaks in different samples. For comparison of multivariate analysis, we have to make the peak aligns among these samples, which means you have to group the features or the peaks from the same compound, ideally from the same compound, same metabolism, together to be able to analyze which the specific compound or metabolite in which sample has a higher peak intensity or higher contents, or in others samples which have less amount. This is the goal. So we developed a program called GIPMA. We just published like last month. So this is the procedure. How do we do the so-called global intensity guided peak matching and alignment? So I will not go into too much details. How do we do the automatic peak matching and alignment? So one thing I want to emphasize is, in this case, because you do peak matching and alignment, you have to define the tolerance of the chemical shift difference among different samples. So in this software, we develop an intelligent slate. This software can intelligently select the optimal chemical shift tolerance for the peak matching and alignment among different samples. The judgment, or how do we evaluate which is the optimal chemical shift tolerance? We try to maximum the number of valid peak cluster. We define what's the valid peak cluster, which is kind of like the final feature we will use to for the data analysis, for the multivariate analysis. So always in other way, we will think this valid peak cluster will be statistically more possible to be a real signal from the real compounds. That is seen. We do the matching and we test in one of our study is we have here we show we do a HCA analysis of the aligned peaks of 5 different botanical arranging of the dendron herbars. This is the Chinese traditional Chinese method. So the overall the picture or this schedule is kind of like this. We have the spectrum of the tracks. We get the peak list. We track the peak information. Like we use the reconstruction of the spectrum, and we use the GIFMA to align this. You get the regularize the data matrix, and then you can do later the univariate or multivariate analysis. So next step, once you have the regularize the data matrix, you have to do several different things, but this is quite a routine. You do the multivariate analysis, and then you get a so-called model. You have to do the model validation. Where usually you have to do 2 steps. One is cross validation to evaluate the model by itself. If it's it can explain the data really good or not. And another is you do a permutation test, which means you have to kind of like give you the somehow idea if the model is fitted or not. It's kind of thing. And then you set some rules to select the differential variables. There's no from the consistent rules, such as you use VIP, they called variable importance larger than 1.0, or you can choose it 1.2, 1.5. Depends on how strictly you can screen the differential variables. Or the P-core. P-core is kind of like a correlation of the variables you measured with one of the components of the model, and some other this p-test. This is a univariate analysis, just t-test p-value, and also some people use the fold change value, but this is not really or statistically measurement. So anyway, you use different rules to select, not really discover, to select differential variables. And once you have the differential variables, you, based on these variables, you try to identify what kind of what's the different metabolites. So you have to determine the chemical identities. So of course, the data analysis, there's a lot of tricks you have to play, like the scaling. You can use the scaling by UV univariate, kind of like a standardized univariate, or they call the Pareto scaling. You use different kind of scaling, pretty much you will get different differential variables. So for the data analysis of a metabolic study, you have a lot of things to play around, and also have to pay attention to quite make sure, not really make sure. You have to judge if the result you get is reasonable. I can only say if it's reasonable, not really correct or incorrect. Because the later stage, as I showed in the first 2 or 3 slides, you have to do the biological verification, whatever you get from the mathematical analysis. So I show you some examples. How do we do the analysis and the comparison of some traditional Chinese medicines? Here is the spectrum of 6 different botanical origin of this Chinese herb, Dendron herba. So you can see this is the overlay of we took 6 spectrum from different species. This is the overlay of this of the 2D HSC of these 6 species, and you can see it's pretty much very complex. There are thousands of peaks or signals, and then we have to do the analysis according to the procedure I introduced just now. You have to pick peaks and deconvolute the spectrum, get the peak list and the peak volume, etc. And then you have to do the peak alignment and matching and alignment, and produce generate the data matrix for the multivariate analysis. And then you will build the mathematical model and choose the differential variables, and you can determine the chemical ID of the so-called differential metabolites. So for this comparison, we try to find out the classical chemical compounds or compounds in each of these species, because they belong to the they are quite similar, but they are different. We want to find out chemically what's the difference is. So we do a PCA and HCA analysis at the 4th stage. It looks pretty much okay. The sample or the spectrum from the same species, they kind of aggregate pretty close. So which means our data is quite reasonably controlled. So to find out the classical metabolites of each of these species, here I show as an example how to find the classical metabolites of the I call it the DC. This is the dendron, this chrysothoxone. So we choose this and make a pairwise PRSDA analysis of this DC black with all the other 5 species. So the black is the chromosin. Make the PCA analysis with all other 5 species, and we choose and together with the thesis, the univariant thesis, and we choose the differential variables by both the univariant and the multivariant analysis. So for each of this comparison, this pairwise, we have 5 pairs. Comparison, we have a bunch of different differential variables, and we make a intersection of all these difference. So finally, we get the 11 common differential variables. We will assume this 11 common differential variables will be the characteristic, will represent the characteristic metabolites from this specific species. And then with the 11 variables or the spectrum features, we expand this spectrum. You can see quite complex. We try to add, and then next step, we try to determine the identity, the chemical identity of what these peaks stand for. So we make a comparison with the data published. Usually the phytochemical studies, they publish this HSC chemical shift of the compounds found from this species or genus. So we identified 3 compounds as the differential or the characteristic metabolites in this species. Actually, the first one is already recorded in Chinese pharmacopoeia. So here again, for very complex the mixture, the data interpretation is also very challenging. Just now I
can you conclude in next 2 minutes, please?
Okay. Oh. So the challenge of integration of the 1D, but even for 2D is still very challenging. So later on is for the identification. I will not go into the details. How to make the automatically identification of this single compound from the complex, the spectrum of the complex mixture. So that's all. Thank you for listening. Oh, one thing I make an advertisement. Our Chinese magnetic resonance spectral society launched a journal called Magnetic Resonance later. So you are welcome to submit your high quality manuscript to this journal. So it's open access and no publication fee. Thank you.
Thank you very much, Professor Kevin. You know, your work is state of the art. The method you have developed for quality standard based on metabolomics of extremely complex TCM are considered to be very, very useful. I know that some of them have already been used in industries, and we do really appreciate your profound understanding and your excellent work. Thank you very much. Thank you.
Okay, thank you. Bye.
With this, I would like to thank both of us. We would like to thank both speakers from China. They are among the best in the field of the application of spectroscopy in various different disciplines. And I'm sure that their contribution have introduced us to new terminologies and new methods. Thank you very much. Thank you, everyone. Thank you.
Okay. Ani, I will show you the picture as well here. You know the this is more comfort, but the online cannot hear. Okay. And you see, this is the Marcellina hellenica. You know the Helen, who many wars occurred during the history. And this is Marcellina hellenica. The name is coming from Helen. It is a special animal. It lives on the pine trees. I also saw the pine trees in this campus. Especially, it lives on the Pinus britia. Not all Pinus britia. The trees which it loves, he can live, she can live, or she can live. It leaves some problem here. Thank you. It leaves a secretion, then the bee uses this secretion and preparing the pine honey. The pine honey produced in Turkey and in Greece only. 10% is almost produced in Greece and 90% in Turkey. And you know the flower, a bee gets the secretion and prepare the honey. And honey, I wrote here flower honey. If any pollen, more than 45%, the honey is named unifillar and also name it the pollen name. For example,
kindly share your slide.
Honey or acacia honey. In Turkish Food Codex, I want to read this one to you. Honey is natural sweet substance produced by the Apis mellifera bees from the nectar of plants or from segregation of living parts of plants or segregation of plants sucking insects on the living parts of plants, which the bees collect transforms by combining with the specific substances of their own deposit dehydrate, store, and leave the honeycombs to ripen to mature. This is in Turkish word. Honey is used in gastric pain, colds, rare infections, oral ulcer, hypertensions, and against kidney diseases, against chronic digestive system, especially peptic ulcer, against duodenal ulcer, against bacterial gastroenteritis in children. And also, it is already known that it's antiseptic, antimicrobial, osmotic, hydrogen peroxide, and acidity. And it also have nourishing and moisturizing properties. And World Health Forum recommended that, it's written here, honey, a cheap antibiotic. Let's see the economy worldwide. In worldwide, 56 million beehives are available. 1.2 million tons of honey are produced every year. 90% of the world honey imports are realized by about 20 produced countries. Let's see the 20 countries. Number of beehives worldwide. Turkey is the third country. India is the first. And when we come to honey production in tons, Turkey is the second country after China. China is almost 35% of the world's honey producer, and Turkey is the almost 10%. Let's see the honey yield. Turkey is the 10th here. Almost 15 kilograms per hive we produce. But this is a bad slide for me. Turkey is the 22nd country exporting the honey. Why? Because most probably we thought that is adulteration. Then we focused on the adulteration, and I saw in the literature that frequent adulteration in honey, direct or indirect or blending. Blending is okay, it is still healthy. Adulteration, but it's healthy at least. But direct and indirect adulterations are giving many problems. I read many papers on this. The adulterated honey gives the mices, which makes the cancer. And mostly starch syrups are using. And if the starch syrup is used, C4 sugar analysis can detect it. So after that, some people are using inverted syrups or high fructose inulin syrup, date syrup, and the jaggery syrup. These particularly the inverted syrup, beet inverted syrup, cannot detect by the C4 analysis. You see the inulin here. And honey, let's look the overview approach to authenticity of honey. Honey, these are okay. Botanical origin and geographic origin, we can do this. And adulteration, during the processing, many people during the packaging and heating the honey and HMF occurs, and many enzymes are degrades. And sugar addition, direct and indirect, and can we detect some instruments, including the spectroscopic instruments. And there are some markers, depends on the syrup. If you detect these markers, it's also said that the honey is adulterated. But mostly the sugar syrup cannot detect by the many analytical instruments. So what is the people done? Sugar beet is solved, prepare it 1:1 or 2:1 ratio, and invert it with an enzyme. Sorry, invert with an enzyme, or invert. The bees are invert here, and bees get tired. And there is an acidic syrup here, enzyme syrup. Here in enzyme syrup, bees don't tire it. And I saw this paper published in 2013, and the feeding type, if honey feed with the honey, the life of the bee is 27 days. And enzyme invert syrup is the closest to that one. When you use the acid inverted syrups, the lifetime of the bee very decreases. What does this mean? I think the beekeepers know this life dates. So I will talk about the beta fructoferronisas and alpha beta gamma amylases and alpha amylase. In honey, alpha glucosidase and alpha amylase is available rather than two enzymes. There is no enzyme, other enzymes called foreign enzymes. And also alpha amylase can be added from outside, and it's also foreign alpha amylase. Beta fructoferronisase is used to invert the sucrose to fructose and galactose. Then this syrup given to the bee. If this syrup given to the bee and bee prepared honey from this syrup, and beta fructoferronisase also passes to the honey. If beta fructoferronisase activity is tested, so honey can understand its adulteration. So we used here the raffinose. Raffinose is normally not available in honey. So beta fructoferronisase inverts this bond and remove the fructose here. And this is the mellibiose. By the detection of mellibiose by HPLC reflective index detector, this can be understood. We validated this method. Method error is 10 unit, and its RST is 5%. And we advised in Turkey, 20 unit is okay. After than 20 unit, we can say that honey is adulterated. And we can we can also use the stachylos, but then the peaks are coming lately. Okay, you saw here the HPLC chromatogram. And the oligosaccharides also, when invert with the alpha amylase, also occurs. You know the amylose and amylopectin parts of the starch. And the common oligosaccharides after hydrolysis of starch is mostly maltodextrose, isomaltodextrose, maltotetrose, and hexose. We saw in the honey. And this is the easy method by using the HPLC reflective index detector. And only the maltose is given permitted in the Turkish food gathers, but up to 4%. Let's see the foreign alpha amylases in honey. And we know already alpha, beta, gamma amylases are available. These amylases is different in optimal pH and temperature. The presence of beta and gamma amylases are not expected in honey. So if the beta and the gamma amylases are detected in the honey, so it means that it is adulterated. So during the adulteration, beta or gamma amylases can pass to honey, or alternatively, if bee is fed a syrup containing alpha amylase, the bee stores the enzyme by converting alpha amylase to beta amylase. And this is the method we used. And this is the substrate. And beta amylase breaks this bond and produce maltose and 4-nitrophenyl beta-galactopyranoside. by using the another second enzyme, it's converted to chromophore group for the spectrophotometry detection. If not, the HPLC dot is also can be used to detect this substrate part. So we studied and we produced controlled honeys. I will say here honey, actually they are not the honey, but in order to mix anything. MyBeer, Muğla City Beekeepers Association proceeded for us 5 type of samples. B feed with the inverted sugar syrup, B feed with the Time honey produced previous year, B left alone to produce natural honey, B feed with starch-based syrups, and only the sucrose. Then we saw these results, and beta fructofuranose's activity is almost 7 in obtained with inverted syrup, and I will focus on this diastase and the proline contents. The diastase should be more than 8 for a natural honey, and the proline content should be more than 300 milligram per kilogram. And the honey obtained from Time honey, you see this diastase number more than 8, and the proline content more than 300. And this is the previous year's Time honey. It's a similar properties. And naturally obtained honey also, you will see here the appropriate content. Then starch-based syrups also give the same problem. Diastase and the proline amounts are lesser. And starch-based syrup has no diastase previously. And honey obtained with the sucrose, also you will see the diastase and proline amounts lesser. So I am now looking for this honey obtained with the sugar. If from outside any alpha amino acid is added, you can see the diastase number, or you can increase the diastase number 8. So how foreign alpha amino acid can be understood? We have checked for the simple and cheaper methods. It's going to finish. And we, by using this cutoff filters, we get the compounds mixture more than 30 Dalton. So this is the slide. You will see the different alpha amylases. Different gives different chromatograms on GPS column. So we focused on that. From different alpha amylases, we can find the impurities, and from the impurities we can catch the adulteration. And you will see that the tentate more than 30 Dalton molecular weight. It's a similar alpha amylases molecular weights. Then we shifted the STS page and the Western blots. We used the honey samples with high diastase and honey samples with low proline and high diastase. Proline is an amino acid. It's free in honey. So proline and the diastases should be proportional. So if the proline is lesser and there is a high diastase numbers, so we suspected this type of honeys. And we also use the other authentic honeys. And you will see the STS page. We saw some different dots here. And I will pass this one. And we went to the Western blot analysis, and you will see that area I draw down. And there are some peaks also showing with a peak with the antibody of alpha amylase. So another adulteration is stable alpha amylase. And you can heat this honey up to 100 degrees and stay there, for example, 2 hours, the amylase content is not decreases. So the diastase is measured when we kept the honey 90 degree for 2 hours and again measured. If the diastase number is stays stable, it means that's also adulterated with the stable alpha amylase. Okay, this is the mola. Professor Iqbal Chaudhry already saw these places. Very good places. Karate, we also went to Karate Hospital. And this is some parts of from our university. And these are the instruments of our laboratory we established. And I should acknowledge the Beekeepers Association of Muğla, Turkish Beekeepers Center Association. Here and I thank to you to listen me. I borrowed you after lunch.
Casual, so you have a question?
Okay, this presentation does not show you that don't eat the honey. Okay, you purchase and eat money. Okay. Okay. In Turkey, in the food codex, honey codex, there is a labeling also. It's labeled that it is just honey. It's a pollen amount, this one, blah, blah. It's already mentioned on the labels. So this is, I must trust it because the laboratories of agriculture getting some sample, going to the shop and getting the sample and tested it. If does not the same property, it's a big penalty. Okay, there is another option. You can get the honey. You can give to analysis. So we are searching cheap analysis. This is another option.
Okay, thank you very much.
I'm the chairman. I have a short comment. The bee population is decreasing dramatically these days, you see, and we have to take care about bees. Use less chemicals in the environment. And as I'm the second chairman, I would like to ask Sultan Nova to show up. She's the next speaker. She's here. Oh, okay. And maybe your presentation is already on the laptop. I will use the time to introduce you from your CV, which is in the booklet. So she's from the Eurasian National University in Kazakhstan. And, sorry, and please correct me when I. So she completed her undergraduate studies in 1998. She obtained a BSc in 2000, MSc in 2000, and then she changed and continued to for PhD studies. So She obtained a PhD in 2010 in chemical research, a doctor of chemical sciences. Already she was starting her research career on salt-resistant plants. And Sultana is or was member of the International Research Center of Science in Karachi, most of us know very well. And she obtained a grant from the Republic of Kazakhstan in 2003 for young scientists and a scholarship. She was awarded in 2013 with a special medal, laureate of competition of science work. And she continued in the field of bioinorganic chemistry of natural compounds. She has over 170 publications, 18 patents, 5 textbook chapters, and is member of expert groups in this topic. The title is on display, and looking forward, due to the time schedule, we suggest your presentation for 17 minutes. We provide some warning, and then we have some time for discussion. Please start.
Good afternoon, dear colleagues. Let me introduce my research work on title The Chemical Constitution and Biological Activity of Some Galaphids from Flora of Kazakhstan. Many plants, such as galaphids, have not yet received much attention as source of bioactive molecules due to limited popularity. It is known that a significant part of land in Asia and Africa affected by salinization, in which perfectly different galaphid plants grow. About 700 species, including in Central Asia, are known to possess some salinity tolerance. Only few species have been investigated for their antioxidant, antimicrobial, anti-inflammatory, anti-tumor activities. In this regard, the investigation of chemical composition and biological activity of galaphids for their future use in the practical medicine is perspective. Actually, the chemistry of galaphids tend to establish that they may be a source of novel compounds and produce some specific secondary metabolites. In this work, in this slide, shows some different arid region of Republic of Kazakhstan. It is west and mostly south part of country in which grown a lot of galaphid species. This research work started on the international project in TAS, and we are collected around salt affected soil Aral Sea, different species of galaphids. So about 40 species galaphids which belong to the family. Sorry. About 40 species galaphids which belong to different family, Tamaricacea, Zygophyllacea, Chenopodiacea, and etc. So we continue this work in the under Pakistan-Kazakhstan agreement between Ministry of two countries, Education and Science and Technology in 2000. Under this cooperation and the leadership of famous world scientists, the result of this collaborative work were published in the different international journals and obtained 14 patents Republic of Kazakhstan. So the 12 galaphids which belong to the genus Tamarix, Campharosma, Zygophilium, Calidium, we investigated in my research group. Let me present some results of this chemical investigation, of some perspective, perspective galophites. First, about genus Tamarix. This plant very widely distributed in different regions of Europe, Asia, and Africa. So by using different methods of chromatography, we isolate a number of chemical constitution. The structure of these all compounds were established by modern spectroscopy, including 2D NMR, COZ, HMBC, HMQC, and different type of mass spectrometry. So in next slide, present some new metabolite which isolate from Tamarix species. These terpenes belong to the pentacyclic triterpenes, difridolean type. Glycoside of these triterpenes, isotamarixin, show high antioxidant and inhibitory effect of the enzyme alpha-glucosidase and beta-glucuronidase. We also isolate some polyphenolic compounds which belong to the tannins. From a fraction of hydrolyzable tannins, which from fraction of hydrolyzable tannins, we isolate so like these compounds which belong to gallic and ellagic types tannins, and the Tamarix species also contain major amount of flavonoids as well. And in next slide, I will show you. In result, we isolated and identified more than 48 compounds. I found some antioxidant, antibacterial, antifungal, antidiabetic, etc., and other activities. So nowadays, we obtained the temporary analytical regulatory document and make registration and have registration certificate of Ministry of Health of the Republic of Kazakhstan, indicating permission for use one of the species Tamarix in medical practice. Next, galophites belong to the genus of Campharosma, family Chenopodiaceae. The species contain a largest amount of essential oil and polyphenols as well. From Campharosma, 14 new substances, for example, triterpenes, flavonoids, chromones, were isolated. So it is found that the chloroform extract possesses immunomodulate activity, and from this extract, we isolated 3 hormones, and one of them show moderate immunomodulate activity. So, next plant which we investigated, it is plant which contain which belong to the genus Calidium, Chenopodiaceae family. From this genus, 24 substances were isolated, and 12 triterpenes and U. So the different extract possess antibacterial, antifungal, insecticidal, grow regulate activity, and some of the individual triterpenes which belong to olean type have been shown very high antibacterial effect. Here, given some of new metabolites which isolate from a genus Calidium. You can see these terpenes belong to triterpenes, olean derivatives. This is new compound. And these triterpenes shown very high antibacterial activity. So from these three genus, we isolate a large amount of flavonoids, and they are represented as aglycone, it is quercetin and camphorol derivatives, and monoglycosides. The glycosides contain glucose, galactose, and arabinose units in third position of the carbon 3 aglycone. Flavonoids, sorry. Some of the fractions which contain flavonoids show very high antioxidant activity, immunomodulate activity. So from this fraction, we isolate some in individual state flavonoids, which show very high antioxidant activity. It is rhamnazine, and from Campharosma species, we isolate some glycosides of quercetin, which show immunomodulate activity in different concentration. In genus Tamarix also were found some rare flavonoids which contain sulfate, and 3 metabolites were isolated. Isolated from this species. One of them show high antioxidant and inhibitory effect on the enzyme alpha glucosidase. In conclusion, this work is dedicated to the investigation of chemical composition and biologic activity of some galophids as a new source for biologic active metabolites. It was found that galophid species contained a large amount of triterpenes, pentacyclic triterpenes, olean or sun types, polyphenol compounds, flavonoids, phenolic acids, hormones, tannins as well. From investigated species, more than 77 metabolites were isolated. 11 NU, the structure of compounds were established by a complex chemical and spectral techniques. So, and in the presentation, I would like allow to express sincere gratitude to the Patron and Chief ICCBS, President NICIC, Professor Dr. Abdurrahman, and Coordinator General of COMSTECH, Director ICCBS, and ITJ Research Research Institute of Chemistry, University of Karachi, Professor Dr. Muhammad Iqbal Chowdhury, for provide strong cooperation and the Pakistan-Kazakhstan agreement. And so now most of this work were done in this famous World Center, and I would like graduate to COMSTECH organization by every financial support to provide for the research and give a new opportunity for our sciences. And I'm grateful for invitation to organize a committee of the University of Jordan. Thank you for attention.
Yes, thank you for the very nice and interesting talk. We have a few minutes for questions because you're staying very good in time. Please, maybe the young participants, please. Yeah.
Yes.
I have a short question. The plants, they're salt resistant or tolerating. They need salt. They can grow also in other places, salt free, or maybe have a different metabolism, or they require a salty area.
Can you repeat question?
Okay.
we investigate only gala feeds which grown in Kazakhstan. We are not make some research from other country, other place. But it's very good idea. Compare different gala feeds and investigate some chemical composition.
There's one more question. Yeah.
We checked toxicity of this plant. For example, species which belong to Calidium and Campharosma have toxic. Yes, yes, we check different. Yes.
Okay, more questions? Interesting topic. Yeah, please.
Yes, last time, last 5 years, we're trying make some scientist work in polymer science. So we get some substances from this plant and make some composition with different type of gels. And so this applicant can use in medicine for skin diseases.
Okay, sounds interesting. Thanks again for the presentation, and I hand over the call.
Thank you.
Our next speaker is Solomon Derisi, and he is going to talk about structurally unique secondary metabolites from Militia useramineses. I hope I'm right. I'm saying it's correct. SSP useramineses and Derris trifluiata. And Dr. Derisi is a senior lecturer at the Department of Chemistry, University of Nairobi, Kenya. He has a vast research experience in the area of natural products chemistry. He has published more than 60 journal articles in peer-reviewed journals and supervised postgraduate students from different countries like from Cameroon, Congo, Eritrea, Ethiopia, Kenya, Uganda, Tanzania. He served also as the Natural Products Research Network for Eastern and Central Africa, and also he is the program officer from 2009 till 2014. He has also a number of NMR training sessions and workshops during summer schools and the pre-symposium in Sudan, Tanzania, Congo, and several countries. Also, Dr. Derisi is currently the Executive Secretary of the Kenyan Hub of the Pan-African Chemistry Network, and also he is a member of the African Hub of the Royal Society of Chemistry. I think he is getting, you have around 17 minutes plus 3.
Assalamu alaikum and masal here. Thank you very much. My name is Solomon, as you're told. I am a senior lecturer at the Department of Chemistry, University of Nairobi in Kenya. And as you know, Kenya is in East Africa, just below the equator. And Kenya is famous for its national parks. Not honey, for safari, and the big five, you'll find them there. It's one of the most biologically diverse country in terms of biodiversity. And this is where Kenya is. And there are close to 7,000 plant species recorded in Kenya. And the majority of these plants belong to 3 families, the Fabaceae, Posaceae, and Astraceae. And since as a student, a PhD student, together with my colleague Abi, we've been working on the Fabaceae species. So the work I'm going to present will be related to the work we've done on this. And these 7,000 plant species, as you can imagine, they look very different from each other in terms of their morphology, the physical features, etc. This is their outside expression. But in these 7,000 different plant species, there are many different chemically diverse class of compounds. So there is much relationship between biodiversity and chemical diversity. What we see is the morphological features, but inside of the cells of these plants, there are many different types of chemical compounds which can be used as scaffold for the development of many different types of useful chemical compounds. So these are some of the famous known plants which look very different from each other, but from these different plants, several of these useful chemical compounds have been reported and we are using them. This is okay, I don't know what happened. Sorry. I'm sorry, Chair. Yeah. Sorry. So this is just what I picked from this publication that came out in 2011, which says plants represent a rich source of structurally diverse secondary metabolites, which can be exploited in the development of new clinically important compounds. Indeed, due to their biodiversity, medicinal plants represent the largest library of compounds that has ever existed. So what I'm going to discuss with you okay, so sorry. So what I'm going to discuss with you, some of the structurally unique secondary metabolites we have isolated from 2 plants that belong to the Fabaceae family. One of the plants that provided us unique secondary metabolites is this Milicia uzaramensis, subspecies uzaramensis. From this plant, we isolated 12A hydroxyrotenoids. In terms of their structure, their gross structures, these compounds are not anything to talk about. They are known, but what makes them unique is the unique stereochemistry at the 6A, 12A junction. So this provides us compounds with this novel, unique stereochemistry as indicated. The other plant which we studied is Deris trifoliata, mangrove. And from this, among other compounds, we managed to isolate new subclass of flavonoids. One of them known as rotenoloids, which, as we shall see later, has a very unique features that makes it to be a new subclass of flavonoid. And these ones, probably later we will see the structures better. These are spirohomo-oxarotenoids. So I want, first of all, to present you the work we did on Milicia uzaramensis. And from Milicia uzaramensis, we isolated a number of chemical compounds, some of which are novel. But what I'm going to discuss and focus on is on the 12A hydroxyrotenoids we described from this plant. And this is the proton NMR of one of the representative compounds. And what will help us to determine this compound is indeed a 12A hydroxyrotenoids is because of these characteristics peaks in the proton NMR in the aliphatic region. Because of these peaks, together with the corresponding carbon-13 NMR, we determine these compounds to be indeed 12A hydroxyrotenoids. So this is the basic skeleton of 12A hydroxyrotenoids, and these are the specific proton and carbon-13 NMR signals that help us to determine these are indeed 12A hydroxyrotenoids. When you look at these compounds, they have two chiral centers, one at 6A, the other one at 12, two chiral centers. As a result, we should expect to see 4 stereoisomers. So depending on the way this hydrogen and the OH are organized, we can have 4 possible stereoisomers. 2 possibilities where the hydrogen and the OH are cis, the other 2 possibilities where the 2 are trans. So among the 4 possibilities, so this is one of the, let me see if I can remove this, sorry. So one of those is this one where the hydrogen and the OH are cis and the corresponding mirror image. And the other ones are where the 2 are trans to each other and the corresponding mirror image. So these are the 4 possible structures in this basic skeleton. And we can determine whether the BC ring junction is cis or trans based on the chemical shift value of the hydrogen at position 1. When they are cis, the shape of the molecule will not allow this hydrogen and this carbonyl to be in close proximity. But when it is trans, this hydrogen and this carbonyl will be close to each other. So as a result, when it is trans, this hydrogen will experience the deshielding effect of the carbonyl. So as a result of that, these 2 hydrogen will have distinct chemical shift values in the proton NMR. So when it is cis, they resonate around this value. When it is trans, around this. And then when you look at the proton NMR of these protons for the 12A hydroxyrotenoid we reported, we found their chemical shift value is around here. So indicating that the compounds, the 12A hydroxyrotenoid we have are have a trans BC junction. So the proton NMR will not help us distinguish between these two enantiomers. So we have managed to eliminate this. We still have the two possibilities. So in order to do that, we measured their chiral properties. We measured their CD. We measured the CD of the CD of the compounds, and we tried to compare the data we generated with what is available on the literature. On the literature, such type of compounds with this specific stereochemistry are reported from this particular plant, and these compounds have a negative Coulton effect at about 340 nanometers. And then we compared the CD curve of our 12A hydroxy with this. Then what we found that, remember, these ones have a negative Coulton effect at 340 nanometer. But what we found for our 12A 12 hydroxy rotenoids, the CD curve showed that it has an opposite Coulton effect at that particular wavelength, indicating they have a different configuration. So these are the first group of 12A hydroxy rotenoids that are reported with this very specific stereochemistry. That's what makes this particular compounds unique. Of course, we did not just use CD to determine this. In addition to this, we used X-ray. We generate their X-ray to indeed unequivocally determine the configuration of the two substances at 6A and 12A. So with that, so these are the 4 12A hydroxy rotenoids, which we reported from Militia ozaramensis. The other one plant which we studied is Deris trifoliata. Deris trifoliata is a mangrove, a legume. It grows around the Indian Ocean in Mombasa. And you have to really go into the ocean and with gun boots and fighting with scorpions in order to access this plant. And we got a number of different parts of this plant, and we investigated them. And we isolated a number of compounds, but the ones I'm going to discuss are those that are structurally unique. So from this plant, we isolated the usual most ubiquitous proteinoids, degulin and tefrosine, degulin and rotenone. And when you look at the proton NMR of such type of compounds, you will see that we have these CH2s, because of the nature of the chirality of this, appearing as a doublet of a doublet. This one appearing as a doublet of a doublet, and the doublet, this one appearing as a doublet, etc. You can easily identify these compounds to be protenoids. Okay. When we look at some of the other fractions and we generated the proton NMR, we saw something different in terms of their the skeleton, their chemical. They are basically the same, except instead of having such substitution pattern, such chemical shift in the aliphatic region, we found to have two methylines instead of just one methylene, as this one indicates, two methylenes and one CH. This can only happen if this ring is open. When this ring opens up, at this position, you'll have a CH2. So this will be a CH2. This will be a CH2, two methylenes with different substitution patterns. So as a result, we got both the open ring of rotenone as well as degulin, and these are the compounds that we report. And so these compounds, such type of class of flavonoids, have never been reported before. So this is a new subclass of isoflavonoids, and which we named as rotenoids. Okay, and so in total, we managed to isolate 3 of such compounds from Eris trifoliata. Of course, when you have a situation like this, if you have enough data, or it is good to see how they are biosynthesized, speculate their biosynthesis, because when nature synthesizes compounds in a specific systematic way, and you should be able to figure out the biosynthesis of such type of compounds. And okay, I have almost, it's just, okay, good. So we had degulin, and when this ring opens up, you get degulol. When it is methylated, this one, and then when it's hydroxylated at this position, you get this. We have managed to isolate all these compounds from Eris trifoliata. In addition to that, unfortunately, this is hiding something. In addition to that, we managed to isolate this particular fragment. This one, okay? So just suggesting it is indeed possible such type of compounds can be justified, their structure can be justified from biosynthesis. The other compound with unique skeleton which we have isolated are these, what are known as spirohomo-oxarotenoids. These are spiro-oxa compounds. Their skeleton is like that of rotenoids, except they have an additional carbon. That's why these are spirohomo-oxarotenoids. This work is published, the details of which you will be able to get. So let me just acknowledge some people before I finish my talk. Of course, this work is done under the mentorship of Professor Abi, who would have loved to be here, but there's some complications because of that. He was unable. He was my PhD supervisor, mentor, etc. And these are the students who worked on this. Some of them were undergraduate students at that time, now with PhDs. And we work in collaboration with universities in the West, at University of Potsdam and Uppsala University. I know in the subcontinent, you are very familiar with cricket. I know you also love football. So these are some of the most iconic figures of football: Maradona and Messi. Yes, and Maradona inspired this young man, Messi. And because of that, he managed, just like Maradona, he managed to win the World Cup. As much as I love football, my iconic figures, I'm telling you that from the bottom of my heart, are such type of people. And I just want to say thank you very much. I'm really humbled to be in front of you when giving this presentation. I just want to tell Professor Taur Rahman that the center he established is in very, very good hands. And in addition to you, you have mentored so many beautiful young women. Some Atya and many others who can take this institution to a higher level. I would like no. I just want to finish my talk by thanking COMSTECH and the University of Jordan, Shukriya, shukran. Thank you very much. Thank you. Thank you.
Can I talk now? We can have very short questions. Yes, Dr. Amal. Yeah, that's good.
Yes, see, of course, I would have given the bio as a result we have on this compound, but just in the theme of this workshop, which is on application of spectroscopic techniques in structure determination, that's what I focused on. Otherwise, this there is Malaysia, Tephrosia, are known to produce proteinoids, which have very good larvicidal properties. So we have determined the larvicidal activity of these compounds, again, Aedes aegypti and others. In addition, we've also tested these compounds for their antiplasmodial activity. Malaria is a very common problem in Africa. I just focused on the structure part.
Okay. Dr. Iqbal.
Yes, yes, yes. Yes, yes. Yes. Thank you. Thank you.
Ask the question, Dr. Christian.
Looks like your compounds are highly water soluble.
Highly?
Water soluble. Or how you crystallize?
No, they're crystalline in methanol.
In methanol.
Yeah, yeah, yeah. Yeah, they are not water soluble. Yeah. Crystallized like a DCM methanol.
I think we should stop it here. Thank you very much, Dr. Solomon, for the nice presentation. Thank you.
Okay, it's my pleasure to introduce the next speaker, Ibrahim Yantan. I just introduced you. Please correct me if I tell something wrong. Ibrahim. Okay.
Yeah.
Sorry. Right. Okay, I have to train. He obtained a degree in pharmacy and in medical chemistry and a PhD in natural products. He's professor at the University of Malaysia, and he was faculty.
University Kebangsaan Malaysia, UKM.
UKM.
Yeah.
Okay, is the abbreviation. Yep. You were founding dean of UKM, and you are honorary advisor of the Asian Association of School of Pharmacy, and you are honorary member of the Center of Natural Products, Liverpool John Moore University, and he has more than 36 years of experience in chemistry and pharmacological analysis of natural products. And the title is on display, is very interesting.
Thank you, sir. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. I'd like to express my gratitude. Thank you very much to COMSTECH, Prof. Iqbal especially, and also Prof. Amal, Mr. Jordan, for inviting me to participate in this wonderful event. However, my talk won't be in chemistry. Of course, doing natural product, you have to do chemistry. But my present work is more very serious problem we just faced recently, pandemic COVID-19. So I'm looking for natural immunomodulators, with dual targeting effects as antiviral, killing the virus, and also at the same time to inhibit the cytokine storm that cause hyperinflammation. So my talk will be into two parts. I will explain COVID-19 has slowed down, but it's still there, especially due to the emergence of the Omicron subvariant from US, about XBB 1.5, and also from China, the Omicron BF.7, and also possibly more subvariants, mutants to emerge. This is cause of concern, or in fact we call it virus of concern, and hopefully there won't be any more pandemic. So, we don't need to use masks and so on. So during the pandemic, a lot of the effort have been done to look for antiviral agent, but it's not easy. So although they use drug repurposing using antiviral, antimicrobial, although with just in vitro data, very little clinical studies, but they use it because it's a state of emergency. So until now, there's no full FDA approval of a specific antiviral treatment for COVID-19. And just a few examples, remdesivir, okay, and also the oral antiviral drugs, the molnupiravir, and also the one also being used widely in Malaysia, the Paxlovid. It's a mixture of two antiviral agents. They are given EUA, emergency use authorization status during the pandemic. Okay, so what's happening now? In fact, during the pandemic, a lot of work being done, but mainly on virtual screening of pathochemicals, which have previously been shown promising results with the previous coronavirus, the MERS-CoV and the SARS-CoV-1. And also some in vitro antiviral study or assays on the pathochemicals against clinically isolated SARS-CoV-2 and some in vivo, but in fact, still not much being done yet. In vivo studies on the antiviral effects of pathochemicals against the virus, infected animal models. And of course, there's not yet any work done using human, but there's no clinical study on mature products of anti-SARS-CoV-2. So let us look at the how the virus damage our body, cause COVID-19. So what happened when the virus or the body, the host cell is exposed to the virus? So our first line of defense, the innate immune system, act as a first line of defense. They sense the virus through the pattern recognition receptors, and also by activating inflammatory pathways. So this way, they remove the virus, get rid of the virus. So, but in a situation, if the innate immune system fails to protect our body, then this will lead to what we say COVID-19, a syndrome of viral replication in concert with the host inflammatory response. So what happens in viral replication in a host? So first, the virus will interact with the receptors, the ACE2 receptors on the surface, and then it will followed by the release of the virus genome, and then followed by translation, proteolysis, and then replication of the RNA by the enzymes, protease enzymes, followed by transcription and replication of viral genome, and then finally there will be assembly of the code, viral assembly. Finally, the virus will be released, and this will continue. So we need antiviral agent to stop the replication. And besides viral replication, we have another problem, the cytokine storm. The body of the host or the person infected the virus, they have hyperinflammation. So due to dysregulated inflammatory pathway, elevation of systemic pro-inflammatory cytokines and biomarkers leading to cytokine storm. So if you look the now you look at the diagram here, there are several targets that plant chemicals or any substances. Oh, sorry, thank you. That can be used. I guess I mentioned about how the virus enter the host cell. So you can use, for example, chloroquine and hydroxychloroquine can be used to inhibit the fusion of the virus to the host cell. So it inhibits the membrane fusion. Or you can have, for example, chromostat mislate inhibits the TMRS2, which is involved in the entry mechanism. It stands for transmembrane protease, protease 2. And then it can also inhibit the protease enzymes. Yeah, the RDRP stands for the RNA-dependent RNA polymerase, or it can inhibit also the other polymerase, example, the main protease, the 3CL2, and so on. So natural products or natural immunomodulators can be targeted for all these bioactive or receptor sites, potential antiviral targets. There's different steps of SARS-2 replication, ranging from receptor binding, entry, and fusion replication. It can also act on the mechanism leading to the cytokine storm. Potential anti-inflammatory target, immunoglobulin-based immunomodulatory drugs that can inhibit pathogenic therapeutics by inhibiting the cytokine storm. You have, for example, like tocilizumab or sarilumab. This will inhibit the interleukin-6 receptor or interleukin-6 signaling pathway. So these are the mechanisms of antiviral effects. You have inhibition of the especially the S protein, ACE2 receptor interaction. Then you have the inhibition of the protease enzyme, and also inhibition of the fusion of the virus. These are some drugs, or sorry, some phytochemicals that have been shown by in vivo studies. They, for example, glycyrrhizine, betulinic acid, they inhibit the SAC2 interaction, entry level, the host and receptor, and the S protein of the virus. It can also inhibit the RDRP, RNA polymerase, for example, curcumin, valcadein, pilarine, and so on, and resveratrol. And then can also inhibit the main protease, the 3CL2, the myostatin, and so on. Most of them are polyphenols. So the entry mechanisms and pathogenesis of the virus, the COVID-19 virus in host cells. And these are the possible target sites. So these are structures of the compounds I mentioned just now. They are alkaloids, terpenoids, and flavonoids, most of them. So I can't see any SAR yet, because not many of them are dated structurally. So it's good if you maybe synthesize analogs and derivative of them and study the SAR for the antiviral activity. Yeah, this is the dysregulated pathway in COVID-19 of the host cells. So you have what happened in COVID-19? The inflammatory pathway will be dysregulated, leading to hyper or high level excretion of pro-inflammatory cytokines and the biochemical biomarkers, the pro-inflammatory biomarkers, leading to hyperinflammation, okay, or inflammation. So these are the responsible or those involved in this related inflammatory pathway, the receptors, inflammatory receptors and mediators like toll-like receptor, TNF-alpha, the interferon gamma, and so on, and the interleukin-6 beta receptor. Or you have, and also these inflammatory cytokines and biochemical markers, the interleukins, especially 2, interleukin-6, interleukin-10, and so on. These are responsible mainly for producing the cytokine storm. And then the pathway involved, the JAK and STAT pathway and F-kappa B pathway. So you look at this diagram, these are the dysregulated inflammatory pathway, and then these are the receptors. So if you can have some, or if you, your natural immunomodulators can inhibit these receptors, in a way you can inhibit the cytokine storm. You can, for example, these are the pathway, the JAK, the STAT pathway. So you have a few of the compounds, a few, in fact, still some of them still under clinical trial, which can inhibit the JAK and STAT pathway. Okay. These are the, again, these are the virus, yeah. So they will mentioned just now, SARS-CoV-2, they will increase the 1-kappa B kinase enzyme degradation, leading to activation of NF-kappa B signaling pathway and inducing release of the interleukin-6 and TNF-alpha. And will not only on one receptors or not in one pathway, multi-pathway or multi-receptors will be involved. So finally leading to cytokine storm, so increased level of all this. And these are the potential targets, drug targets for anti-inflammatory activity. So if you look at different stages of COVID-19, the stage 3 of COVID-19 is one. That is, we say the beginning of the serious effects of COVID-19, the inflammation effect. This is at stage 3 of the COVID-19 symptoms. You have the cytokine storm, or we call it cytokine release syndrome, CRS, or hypercytokinemia. Okay, so there will be increased exaggerated levels of the pro-inflammatory cytokines and biomarkers of inflammation, leading to rapid onset of the acute respiratory distress syndrome, and this will lead to death, or lead to organ failure and finally death. So COVID-19 patients exhibit hyperinflammation, an immune system triggered by the virus. So you need an immunosuppressant, which down-regulate inflammatory responses and reduce the excessive cytokine production associated with respiratory viral infection. So then the combined use immunosuppressant then can be used as an adjunct or adjunctive therapy to reduce the cytokine storm with an antiviral agent to provide supportive treatment for patients with COVID-19. So you can use as an adjuvant with antiviral agent. One is to kill, dual target. One is to kill the virus, and one is to inhibit the inflammation, the cytokine storm. So there's very little information on the immunological effects of plants. It's been used traditionally for treatment of various ailments related to the immune system. Yeah, maybe we need to translate the ethnopharmacological knowledge of plants being used to treat immune-related diseases. So very little information on that. So many therapeutic effects of plants extract may be due to a wide array of immunomodulatory effects and influence the immune system. So phytochemicals such polyphenol, flavanol, and so on, they may be responsible. In fact, some have been reported to be responsible for the plant's immunomodulating. And herbal medicine, which is in fact a complex mixture of compounds, they can be used as a multi-component agents to modulate the complex immune system in the prevention of infections rather than treating the immune-related diseases. if you look at how phytochemicals works, it's not straightforward. People are just looking at the reductionist approach. One compound acting on one receptor, producing one pharmacological response. You are talking about reductionist pharmacology. But if we are looking at plant extracts, they are multi-component. So you cannot use reductionist approach. It's multi-component, interacting with multi-receptors, producing multi-effects. So, for example, phytochemicals, they can act directly as antioxidants, as anti-cancer, act on tumor cells, stimulate apoptosis, or they can act antimicrobial by inhibit the cell wall synthesis, inhibit protein, nucleic acid synthesis, and on. But remember, it's also acting on the immune system. It can suppress, it can stimulate. When it's suppressed, the reactive host species will be suppressed, and this will lead to anti-inflammatory, anti-cancer via the inhibition of signaling pathways. It can also, if it's inhibiting, if it's stimulating, so well, it can be used as anti-cancer, yeah, because it will stimulate ROS in the tumor cells, leading to apoptosis. Or it can also kill the virus or the microbes through the increase of the ROS as a defense or killing mechanism. So you can see the effects is complex. It's multi-target. It's not only acting on one receptor. So this is just to show you the pathway involved. Example of polyphenols targeting of signaling reduction pathway as an attractive therapeutic approach for development of anti-inflammatory drugs to prevent any pathological consequences. This is what I have discussed just now. There are many receptors here, the interleukin-1 receptors, the TNF-alpha, yeah, which are responsible for the release of the pro-inflammatory cytokines. So if you can target this block, the block markers here, it shows the spot where you can inhibit the pathway and directly will prevent the release, the unregulated release of the inflammatory or pro-inflammatory cytokines or biochemical markers. I still have time? Two more minutes. Okay. These are examples of drugs. A lot of work being done of phytochemicals. Some of them are in clinical studies, clinical trial stage. Yeah. Of course, curcumin, resveratrol, yeah, capsaicin, and so. And the one that is of interest is andrograpolite. This during the pandemic, andrograpolite was much highlighted in clinical study to show its potential as an antiviral agent and also anti-inflammatory. So, the phytochemicals, many polyphenols with immunomodulating anti-inflammatory activities might find therapeutic use as suppressants targeting the cytokine storm in COVID-19. So this example, yeah, of polyphenols and other compounds that has potential. And these are some research data of curcumin, of eugenol, of resveratrol in relation to COVID-19 suppression of COVID-19 symptoms. For example, eugenol found in glove, in fact, was widely, especially in social media, people saying that glove can be used to treat COVID-19. There's a reason for that. It contains eugenol, which downregulate or inhibit the pro-inflammatory cytokine that can reduce the leukocyte recruitment in the lung tissue, and also it's a natural immunosuppressant that can be used as an adjuvant along with antiviral agent. So it's a dual target. Besides antiviral effect, it can also suppress the cytokine, the hypercytokinemia or the cytokine storm. And resveratrol, very widely investigated for its immunomodulatory activity, anti-inflammatory, antioxidant activity. Also, it shows some promise, yeah, because it inhibits the NF-kappa B activation by inhibition the 1KB kinase and also expression of the COX-2 and inos in cytokine-stimulated human primary airway epithelial cells. So they, for example, resveratrol has clinical trials, 92 studies or clinical trials being carried out. 27 studies are linked with its anti-inflammatory properties. So andrograpolite, this one's there's dual target. effects reduce the especially the dysregulated inflammatory pathway. As mentioned just now, it will reduce the NF-kappa B pathway, activation of NF-kappa B pathway, levels of the MAPK pathway, and also ongoing clinical trial is what I mentioned earlier, combination of Helicobacter canuclata and Eleutherococcus centicosus. This, they call it Kanjang, is a formulation. You can provide scientific evidence on the effectiveness of Kanjang treatment. But however, the clinical study is still preliminary because the sample size is small, and also the study is not really systematic. They don't have phase 1, phase 2, phase 3. They just have one sampling data and look at the effects. They have not really studied the toxicity and so on. But it shows some promise. Yes. Maybe 1 or 2 minutes. I'd like to share with you the methods that we use for antiviral activity and also anti-inflammatory activity, working on the SARS-2 virus, single cell, and also in vivo animal being infected with the virus. So we do virtual screening of natural immunomodulators based on our previous work. So we have here identified around 65 very potent immunomodulating activity, mainly immunosuppressant. And we do virtual screening to look at the spike protein of the virus and the interaction with the main protease, and also with the RPRD, the RNA polymerase. This is the virtual screening, and then followed by molecular docking, molecular dynamic simulation to look using various software models, docking simulation using graphical user interface program. And then in vitro antiviral activity, you use the E6 cells. This is African green monkey kidney epithelial cells. We use the cytostatic effect, CPE inhibition assay, to inhibit lysis of the whole cells and ability of the virus to reproduce. Okay, and then the plaque reduction assay and visualization under transmission electron microscope. This is for in vitro antiviral activity. And of course, we have to run it in this laboratory with the biosafety level 3. So you can see students, researchers have to wear proper attire to the experiment. And then we have BCL level 3. And then for the in vivo anti-inflammatory effects, we use the MHVA56 induced surrogate mouse model of COVID-19. Forming similar hyperinflammation and pathological damage caused by SARS-CoV. Of course, this is all based on previous work, this data, published especially by the Chinese scientists. They have it advanced using Chinese traditional medicine to look at the ability of their medications, the traditional Chinese medicine, to treat COVID-19. So for the in vitro anti-inflammatory activity, we use HUH7 cell. This is human hepatocellular carcinoma cell lines. So we look at the key protein expression, the pro-inflammatory cytokines, and also biomarkers by Western blot assay. And then we use the RT-PCR to look at gene expression. Oops. The previous slide on the flow of activity. Yeah, I think I don't have time. I just show you very fast. This is a virus isolate, and we use the subvariant, the most recent one, the XBB 1.5, and also the BF.7, and also we compare with Delta, because Delta almost very voracious, yeah. So to see the effectiveness, and this is a plant sample compounds, and this is the HU7, is anti-inflammatory activity, and then we look at the use Western blot to look ELISA and so on, and then you RT-PCR. conclusion, of course, still ongoing. I got a 2 years project from the government on this. Hopefully, will be very interesting research outputs for us to continue doing. Because you know it's not easy, especially you're working with a very dangerous sample like virus COVID-19. You need to have proper facility, and then from our recent work, we have identified these 3 compounds. Yeah, the endographolite. Yeah, endographolite. The from Androcarpus paniculata. Yeah, the natural. These are the most potent. If you look at the IC50, the antibody is 0.034. The natural immunomodulators with strong antibody effects against the virus and anti-inflammatory effects against the this-related inflammatory signal of the virus vector cells will provide lead molecule which have potential to further develop into drug candidates for treatment and management of COVID-19. And also discovery of potent antiviral agent with dual targeting ability. Besides killing the virus, it also inhibits the cytokine storm. And this will lead to development of promising agent for treatment of COVID-19. Thank you very much.
Thank you. It was a very interesting and important topic. It's certainly timely. The next virus may come. And I know myself, I was also working in a BSL-3 lab, so it's not easy. Working and going in and out is also a procedure. Questions, please?
Yeah. Thank you. Omen, welcome. Yeah, yeah. True. Yeah, the source doesn't matter here because the most important thing, I want to look at the chemistry, the structure, and then hopefully later on, I want to work on the SAR, structure activity, because this is just preliminary. More work needs to be done. Resveratrol is a very good model, yeah, because it's been shown to be very strong anti-inflammatory activity. So my hope, and also from previous work, it has some antiviral effect, but not against the COVID-19 virus. Yeah, yeah. But of course, later on when you develop, you don't put resveratrol as the compound. It's just the candidate or starting molecule. And then what you need to do more SAR. You want to get some information how. Yeah. 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 But I have seen the wrong message that is being maybe due to their understanding. People are taking, let's say when we say resveratrol is good, you know, people start taking clove, people start taking grapes. That is not right. We look at the effects, they have different stage mechanism. For example, when somebody just got it, the virus, you need to have an antiviral agent. You cannot take right away an anti-inflammatory. It will severe effect, right? So after stage 3, when there's inflammation, then you go for anti-inflammatory. So that's the reason why you need advice professional, medical doctor, and so on. Sometimes medical doctor, you can see also spreading false information about it without understanding the mechanism of action. So we need natural product chemists, medicinal chemists to provide the right information to them. Thank you. Okay. Why? Okay. Because yeah, just means. But I'm promoting peanut because I don't like eat it with alcohol. Sorry. But it's okay because peanut also contain a lot of alcohol. No big deal.
I guess we can just final question.
Yeah, yeah. Of the virus. Oh, the image is already being known. Yeah. And also I follow, well, I draw structure based on the real photo, no, myself, no, there's a software. Yeah. 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 Yeah, use it DM. Okay, thank you.
Thank you again. And the next speaker is Nick Ziti Amina. I hope I pronounced properly. And in the booklet, I'm not seeing a CV. Maybe you can compliment. She's from the Department of Chemistry, Faculty of Science Technology, University Airlangga, Indonesia.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. First of all, I would like to say thank you to Professor Iqbal and also Professor Amal for invitation today. I would like to present, I think it's different with all of the presenter before, because I would like to introduce our group research. The title, Biotentrop Research Group Profile Collaboration Opportunities. I'm Nani Siti Aminah from the Department of Chemistry, Faculty of Science and Technology, Universitas Airlangga, Indonesia. First, I would like to introduce where is Indonesia. Yeah, this is our country, Indonesia. From this until I think in this also still have the island. Professor Ibrahim Jantan country in here. And part of the island, this one and this one is also Professor Ibrahim country. So we are very, very near as neighbors. Yeah, we have 17,000 islands and also 270 million people. So we have more than 3,000 languages, but all of us have the same language name in Indonesia language. When I see from the Google, the position of Indonesia, my city in Surabaya, just 9,000 kilometers from Aman. But really, I came here to 24 hours because the transit 3 times. I have to go first from Surabaya, our city, to go to the capital city to Jakarta, and from Jakarta we have to go to Doha, and then finally we visit Amman yesterday. Very nice city. Yeah. Yeah. Our university in the east of Java in Surabaya. Here is the city of Surabaya. Surabaya come from the name of the fish that named Sura, and Baya is the Java language of the crocodile that our city name is Surabaya. So our university have 3 campuses, A campus, B campus, and also C campus. We are in the east of Surabaya. The A campus is the old campus, now maybe 100 years. And then the B campus is the second new campus, and our C campus is the newest campus where our faculty of Science and Technology in there. Professor Iqbal is one of our young professor in our university. Okay, this is the profile of our group research. The leader is Professor Josephine Wulanwanuhara from the biology. She worked in plant tissue culture and plant biotechnology. We are in the same age. Me, Nani Siti Aminah, and Dr. Alvinda work in natural product chemistry. Professor Heri Purno Basuki, biology, structure, and development of the plant. And we have 4 doctors, young people that work in, we joined together on 2021. So our vision of the group research is to be research center that is able to contribute to provide basic information and applied technology about the potential of Indonesia tropical medicinal plant as raw material for herbal and pharmaceutical industry. So we try to identify, explore, and conservation of Indonesia tropical medicinal plant, in vitro culture and potential medicinal plant, and some other work in our research group. For our research group topic is exploration of the secondary metabolite compound from Indonesia tropical medicinal plant, in vitro culture of potential medicinal plant in bioreactors, integrated microbiome and metabolomic analysis of Indonesian medicinal plant to identify the key factor of plant microbiome interaction. So for the exploration, we do isolation and structure elucidation that all of us as natural product chemists work for the secondary metabolite compound, especially from Indonesia plant. Now we already have isolated 20 species of the Indonesia plant. We also want to carry out of the activated test of the secondary metabolite compound, but still in antioxidant activity using TPP8 or something like that, and also toxicity using PSLT, cytotoxicity using cell line, antimicrobial, and also antifungal activity. We also try to develop the nanoherbal from extract of Indonesian medicinal plant. I think this is a traditional work that selected plant, we do extraction and then isolation by chromatography, and the pure compound, we try to check the in silico and in vitro and also in vivo test. We also make the pure compound and also extract to be nano encapsulation. In our dream that next we can find the nano supplement or nano cosmetic, but now still working hard. This is the higher plan that we also work, and we also done the taper, and in Java language we call it the polok kependem. Kependem mean in the soil, that if we want to find this type of the taper, we have to open the soil and then we will find it. And also we try to explore the Indonesian herbs. This is such kind of Indonesian herbs that we already collected. Now we have the collection 21 of the Indonesian herbs. One of them is the ginger, Professor Rafat, that we discussed together, that maybe in the next we can continue for the collaboration about the ginger from the Indonesia. And then some other this is all of the Indonesia herbs that we already collected and to try to do for our research. For the cell culture, we try to production of the biomass and bioactive compound from potential Indonesia tropical medicinal plant in a small bioreactor that we built in our lab, and we try to biomass and flavonoid production of the Ginura procumbens to different type of bioreactor and enhance of the bioactive compound by elicitation and other treatment. We also check the biomass on saponin production of Thalinum paniculatum. So this is the bioreactor that we already put in our faculty and our research group. We try to produce the root using this bioreactor, also the leaf of the species that selected species. This is our kind of the work that by Dr. Anyar, that he tried to find the wild plant from the place in Indonesia to check the microbiome and to see the interaction of the key factor in plant microbiome interaction. So the activity test bioactive compound using in vitro cell culture in vivo. We do antibacterial, anti-candidal activity of selected Indonesia medicinal plant, antioxidant, especially in Ginura precumben, and we also check the anti-cancer of the Ginura, and also we try to find the potency of bioactive compound of Indonesia plant as immunomodulator. So when I was young, usually my mother prepare for the breakfast using such kind of this tabur. But now in Indonesia, in the morning we eat rice, in the for the lunch we eat rice, and the afternoon we in the dinner we also eat rice. So this is really is very good food, I think, because this the index of glycemic is lower than rice, but little bit difficult. We try to explore the chemical metabolic secondary of the and then we find some phenolic compound from this kind of the tabur. From Dioscorea alata, we got the 3 phenolic compound, that one of the similar with resveratrol, but the position of OH in here, Professor Ibrahim Jantan. At resveratrol, the position of hydroxy in this position, I think. Yeah, this is also another type of Dioscorea that easily, when I was young, this is easily find in near our house, but now the people don't want to plantation of this kind of the tabur. This is, I think, the sweet potato, Ipumo patatas. We have many type of this. The color is yellow, sometimes is violet, and then also white. We try to find that the major component is scopoletin and also amyl parahydroxycinamate. Gambir is one of our product in Indonesia that exported until now. In the world, 80% come from Indonesia, but our people in Indonesia exported in this part. We know well that gambir have a high concentrate of the catechin. We try to isolate the catechin. we have 1.8 percentage of the catechin. When I was young also, maybe now I'm so old, the people use this kambir as like this, that they, to make the mouth and can to make this antioxidant, I think. Yeah. And finally, that we try to isolate the catechin that the concentration is very high. So from this catechin, we continue our isolation, and because of the concentration is high, that and easy to isolate, one after extraction and one time of column, that we get the catechin. So we try to make it small synthesize by esterification, that after esterific we have the ester, that we try to check the IC50 of the catechin and the synthesize, that the synthesized compound have higher activity comparing the catechin. And then we also try to make nano encapsulation of the extract of the kambir. We check it for the chemopreventive using mice, and then it's very active. Also in vitro test. So just a little comparing Professor Iqbal work and another professor, that we try to get the patent. Now one is already granted and another now in process. So the conclusion is, in my presentation, is we want to get more collaboration. So we already have the collaboration with Professor Yoshiaki Takaya from Meiji University. Of course, Professor Iqbal, and the first time that you visit to our university, this is the picture. And then we have also from UPM, from Myanmar, and also from Nigeria as our postdoc by online. So hopefully we will have next collaborator for joint research and publication, student and staff exchange program, community services, and other. Before I close my presentation, I would like to say thank you for COMSTECH for some activities together with Universitas Erlangga, and so for Jordan University for inviting me. Hopefully we will have the new collaboration in the future. Universitas Erlangga that have fund for us from 2003, ICCPS for our work, some of the students in ICCPS, from Directorate of Higher Education of Indonesia for funding research, Faculty of Pharmacy, Meiji University, Biotechnology of Tropical Medicinal Plant Research Group, Universitas Erlangga, and Bioinformatics Laboratory. This is our student, our team that work together to find some exploration, identification until synthesis, also the activity. Let's work together to get the big success. Thank you very much. Sukron. Terima kasih.
Yeah, thank you for the very nice presentation, and you look very dynamic for this long trip. And yeah, and emphasizing again, natural chemistry is important.
Yeah. Yeah. Yeah, in Malaysia. Yeah. Yeah. One of the, yeah. maybe because we only send the Gambir raw material. That's why it is. Mhm. Yeah, yeah, that's for that. Uh-huh. Mhm. Yeah, but really the antioxidant is very high. Yeah, yeah, yeah.
More questions? Final question? No? Then thank you very much again for the presentation. And the last speaker of this afternoon session is Begum Rokia. Oh, she's coming from the University of Health Sciences in Bangladesh. And we'll briefly introduce you. So Begum Rokia is professor and head of the Department of Pharmacology, Dean of Basic Sciences Faculty, and she completed her PhD in pharmacology at an interesting place these days, Institute of Pharmacology in Kiev, Ukraine. And she has a tremendous experience in the area of natural products. She published over 95 articles, and she has contributed or has 6 book chapters prepared. And she's member of the Physical and Pharmacological Society in Bangladesh. And I guess, yes, it's in time. The first slide and title is on display.
Assalamu alaikum, ladies and gentlemen. I feel really honored and privileged to join this workshop here in Amman. And I would like to extend my sincere gratitude to the organizing committee, especially COMSTECH, Professor Iqbal Chaudhary, and also University of Jordan, Professor Amal Aboudi, for inviting me to join here. So as you have heard, I'm going to talk on the phytochemical studies and evaluation of the antidiabetic activity of different extracts from some edible plants in rodent model of diabetes. As you have seen, diabetes has become one of the most challenging public health problems of the 21st century. Why? Because diabetes is not a single disease. Rather, it is a syndrome with the disturbances of carbohydrate, protein, and fat metabolism, with one cardinal finding, that is hyperglycemia, means raised glucose, raised blood glucose. So, and despite the great strides that have been made to understand the disease, the etiopathogenesis of the disease and the disease-related complications are increasing unabated. As we see that the IDF, International Diabetic Federation Atlas 10th edition, states that diabetes around the world in 2021 will be 537 million adults 20 to 79 years are living with diabetes, 1 in 10. This number is predicted to rise to 643 million by 2030 and 783 million by 2045. So the problem is pure. And what is happening in Southeast Asia? 1 in 11 adults are living with diabetes. The number of adults with diabetes is expected to reach 103 million by 2030 and 151 million by 2045. Over 1 in 2 adults living with diabetes are undiagnosed, and there are 747,000 deaths caused by diabetes in 2021 and $10 billion spent on diabetes in 2021. So in Bangladesh, the scenario is also very hectic. We have 8.4 million people with diabetes and another 8.4 people have pre-diabetes. That is, diabetes is knocking at the door to those people. So, and this number is going to increase in the upcoming years. And the WHO and IDF predicts that Bangladesh will occupy 10th position over the world. So that is a big problem. Whenever we talk about the diabetes, when we talk about type 1 diabetes, we know that there is an absolute deficiency of insulin. So we know what is the, we need to, for type 1 diabetic survival, we need to give insulin from outside. But what happens when there is type 2 diabetes? In type 2 diabetes, it's the pathogen is very complicated. It resembles like a mosaic. It means that there may be hepatic insulin output raised, there may be insulin deficiency, there may be post-meal insulin, rapid insulin release lost, there may be fasting hyperinsulinemia, and there may be insulin resistance. What is insulin resistance? Probably you all know. There is too much insulin, but that insulin is not working. There may be hypoinsulinemia, means there is no insulin to cope up with the glucose. So this is omnious octet. These 8 organs actually responsible to produce the hyperglycemia that we found in diabetes. And we all know that there are so many complications. Uncontrolled diabetes, there may be adult blindness, there may be kidney failure, end-stage renal failure, which is usually eventually ends up with dialysis. There may be gangrene, there may be neuropathy, heart attacks, and stroke. And let's see what are the existing oral therapies nowadays for treating type 2 diabetes. Now, one thing I forgot to mention that the people with type 2 diabetes is the highest globally. Only the prevalence of type 1 is prevalent in Scandinavian countries, but other parts of the world we have type 2 diabetes, majority of the diabetic population. And these are the preparations which we have. We have biguanide, we have insulin secretagogues like sulfonylurea, we have alpha glucosidase inhibitors, we have thiazolidine and their derivatives, and we have incretin-based therapies and like injectable incretin mimetics, and we have incretin enhancers, but these are expensive drugs. Moreover, I want some water. And moreover, the medication that I have told, that I have discussed, they have severe limitations. Majority of these medications have significant adverse effects. So we need to focus on more medications that will eventually address the defects, because the diabetes have multiple abnormalities in diverse tissues, as I have told. So we should look for alternate strategies. The enormous cost for modern treatment indicates that alternative strategies are desperately needed, and plant materials are being used from time immemorial to treat diabetes. I was really amazed today from Professor Ataur Rahman's lectures that he told that metformin, this is the best, this is the highest prescribed drug nowadays throughout the world for treating type 2 diabetes. And he said that this metformin has got some anti-aging effects. That is, I was really amazed to know. And you know that from where this metformin came? It came from Galegia officinalis, from plant source. So with the find out a new leaf, from plant sources. We have been started working with BARDEM and Dhaka University. They had been our counterpart. And in BARDEM, we had been working on trying the biological activities of the plant extracts on different type of animal models. So the models were non-diabetic rats, type 1 and type 2 diabetic model rats. But to screen only one plant extract in singles, only on type 1 or only on type 2 or only on normal rats actually doesn't tell you that whole story. To see the profile, you have to do all the 3 models, normal, type 1, type 2, and 3 prandial states. Means in the fasting condition, postprandially. Postprandially, again, you have to, sometimes you know that doctors prescribe them anti-diabetic medicine. He or she say that you should eat with food. Sometimes they say you should take it in an empty stomach. So we also, while working with plant extracts, we actually worked on the animal models which have got these and screened in these 3 prandial states. Altogether, 9 series of experiments we have to perform to screen only a single plant extract. So our this approach actually tells us which part of the plant is active, which prandial stage the plant extract is active, and which animal model. So actually, this methodology helps in designing future biological experiments to investigate the mode of action of this material in greater detail. So, following this experimental approach, we have screened a number of plants from Bangladesh, India, Pakistan, Nepal, Sri Lanka, China, and Cameroon. And we found that, as Professor Dantan was telling, that while you are working with plant extract, you cannot pinpoint single property. Then you might have antioxidant activity, you might have anti-diabetic activity. So while screening, we found plants we have got insulin enhancing insulin secretion, which delay glucose absorption, plants acting on peripheral tissues, plants with mixed activity, plants with which prevent all damage of beta cells or help in regenerate beta cells. Then plants with antioxidant activity, plants with anti-inflammatory activity, and plants with lipid lowering activity. So I'll now say a few words about some plants which actually show delayed glucose absorption in the gut. Because we know that postprandial hyperglycemia is a special problem in diabetes mellitus, because we now recognize that normalizing postprandial blood glucose is more difficult than normalizing fasting glucose. A diabetic might have impaired fasting glucose, a diabetic have impaired postprandial blood glucose, and a diabetic have both. So, but it is recognized that normalizing postprandial blood glucose is very much difficult. So during screening, we found the following plants. There is a number of plants which actually delay glucose absorption in the gut. Why this technique is important, I'll explain. This technique actually important because the glucose level will be, the glucose will be absorbed, but it will, a diabetic have very rapid glucose absorption, so there's an abrupt increase in glucose level. This technique, delaying glucose absorption technique, actually helps the ill beta cells of the diabetic subjects to cope with the glucose load. So ultimately, it actually lowers the diabetic complications. So we found this Tamarindus indica. This is the fruit we have in our country in Asian region, and this Tamaris indica, we found that we see the effect of Tamaris indica on fasting blood glucose levels of type 2 diabetic model rats, and we find that it has no effect on fasting glucose level. But when we studied the effect of this Tamaris indica seed powder on blood glucose, we found it actually significantly lowered blood glucose level when it has been given 30 minutes before glucose load. So we searched, we wanted to know the seed powder showed hypoglycemic effect in the fasting state, no effect in the fasting state, no anti-hypoglycemic effect in type 2 diabetes model rats when fed simultaneously, but showed significant hypoglycemic effect when it was fed 30 minutes before glucose load. So we thought that it might delay glucose absorption in the gut. Therefore, gut perfusion technique was used to assess the activity of this extract. So this is this experiment was done on diabetic model rat. So we have to insert cannula, and on a living rat we have to work on this. So we insert cannula and catheter 40 centimeter apart, and then we actually introduce the extract with solution, and there are lots of things, steps with glucose, without glucose, and the single rat actually serves its own control. So here you can see the rat. This is the, we have just make it unconscious to sleep for 1 hour, and we can perform the experiment. So we found that in normal there was no effect, but in type 2 diabetic rat, it actually suppressed the glucose absorption. So Tamarinus indica powders exerted strong inhibition on glucose absorption when compared with control type 2 rat. So Tamarinus indica seed powder significantly inhibited the gastrointestinal motility when we actually, how it worked, because it decreased the GI motility when we examine through barium meal. So we made the conclusion that Tamarinus indica seeds possesses anti-hypoglycemic properties in type 2 rats, which is at least partly due to its inhibitory effect on intestinal glucose absorption. And this effect cannot be attributed to the acceleration of intestinal transit. So this is the work, also inhibitory effect of Ipomia aquatica. This extract was we got from Cameroon, our under fellow Celestine Soking, and we found that this plant, Ipomia aquatica, delays glucose absorption in the gut. This is the plant Ocimum sanctum. It's a holy basil, it is named. So actually, this plant is it has shown so many activity, increases insulin secretion from the pancreas. It increases glucose store in the form of glycogen in the liver. It actually increases glucose uptake in the muscles and increase carbohydrate absorption through the GI tract. So the results showed that ethanol extract of the leaves of this Ocimum sanctum significant hypoglycemic effect on fasting state and anti-hypoglycemic effect in postprandial state in type 2 diabetic rats. It did not show any effect on type 1. So oral active hypoglycemic effect on normal and type 2 rats in fasting and postprandial states may be due to enhanced insulin releasing activity, and we worked on insulin activity on isolated islets, and we found. And in addition to, there may be interference in the intestinal glucose absorption. So we actually worked with this objective to investigate whether the anti-hypoglycemic effect is related to the inhibition of carbohydrate absorption in the gut. And for that, we gave them sucrose content in 6 segments, namely the stomach, the upper 20 centimeter of the small intestine, then the middle 20 centimeter, and the last portion of the 20 centimeter of the small intestine, and the cecum, the large intestine portion, and cecum. And in these 6 segments, we actually tested the remaining sucrose, and later we, by backup calculation, we made how it is, what is the amount remained in the 6 segments. So we found that ethanol extract of Ocimum sanctum significantly suppressed postprandial hypoglycemia after sucrose ingestion, and increased the residual sucrose content throughout the gut at 2 hours after sucrose ingestion, because we sacrificed the rats in different timing, 2 hours, then 30 minutes after sucrose ingestion, 2 hours, then up to 4 hours, and in different timing, we actually sacrificed the rats and we wanted to see what happening. So we also studied the effect of this Osimum sanctum extract on intestinal enzyme activity. Then extract showed significant inhibition of intestinal disaccharidase activity. You know, these are the brush border enzyme, disaccharidase, maltase, etc. They are responsible for breaking down the complex carbohydrate. So these findings suggest that reduction of sucrose absorption may be partly related to the inhibition of disaccharidase activity in the gut. So these are the results. So the conclusion was that the extract can delay sucrose absorption without suppressing it, which may be due to inhibition on rapid digestion and absorption of carbohydrate at the upper part of the intestine. Undigested carbohydrate is digested and absorbed beyond the upper part almost completely, and this reduction of hyperglycemia by the extract is at least partly related to the retardation of carbohydrate absorption in the gut. This is another plant we have walked, and this is the plant here we found that ethanol extract of this Clerodendron infantulatum showed this was a chronic feeding for 21 days after this experimental period. This ethanol extract significantly reduced glucose level. So we worked on details of this plant, the TLC pattern of this chloroform extract, and this is the TLC ethanol extract of chloroform, Clerodendron infantulatum plant extract. And these are the 5 fractions we got from this. And we went to see whether this fraction actually increased insulin secretion from the perfused pancreas. For that, we have to isolate the pancreas in a living condition and have to keep it for 1 hour with all the measures. And this is a very tedious operation. More than 100 sutures need to keep, and there we have put the living pancreas. Oh. And the fraction collector, the supernatant, we actually measure the amount of insulin liberated through this extract, and we found that to that pancreatic perfusion, we found that fraction, there were 5 fractions, fraction 1, 2, 3, 4, and 5. If you look at the result, we can see that in fraction 2, there was a significant increase in insulin secretion, even in 3 millimolar glucose. We know that 3 millimolar glucose is the base of glucose. Usually it does not actually enhance insulin secretion, but this extract was capable to enhance insulin secretion at 3 millimolar glucose, not talking about the 11 millimolar glucose. 11 millimolar glucose is a stimulator. So we found that fraction 2 was even active in 3 millimolar as well as in 11 millimolar glucose. So we actually wanted to see the structure of this compound by using IR extra proton NMR, and then 13-carbon NMR and mass spectroscopy. And structural studies by spectroscopic method clearly indicated that it is a pheophylline. This is chlorophyllic compound which actually was responsible for insulin secretion. So now there were some plan. We had some work on to see the, diabetes always accompanied with the complications. So we heard, wanted to see the effect of plant materials on diabetes complications. So for example, platelet aggregation. So the effect of Cinnamomum tamala oil, that is eugenol, and Osimum sanctum water extract in vivo and vitro on the aggregation of platelets were studied. And we found that
2 minutes.
Okay, I'm concluding. So we found that Cinnamomum tamala and Osimum sanctum both seems to have potential antiplatelet activity on type 2 diabetic model rats. There is antiplatelet components in the non-saponified part of the oil extract of Cinnamonum tamala. The detailed dose response and mechanism of action of diffraction need to be worked out. The compounds are yet to be isolated and characterized. So, as a result of our outreaching activities, we have published a book with the results of 67 edible plants. So, and there we have mentioned how to eat, what time the plants might be helpful for the diabetic people. So the concluding remarks is that type 2 diabetes is a disease of enormous consequences to the health and wealth of modern and developing societies. All avenues must be explored to provide the most efficacious and safe medicines for the long-term therapy of this chronic condition. Here we have presented some role of plant-based natural products with their beneficial properties on animal models of diabetes as an evidence of anti-diabetic agent that might be helpful to develop new drugs for the treatment of type 2 diabetes and its complications. So they are our mentors and co-workers. And I must acknowledge our sponsors: International Program in the Chemical Sciences, International Foundation for Sciences, UNRWA, Diabetic Association of Bangladesh, Bangladesh University of Health Sciences, where I belong, and Dhaka University. Oh, this is the thank you slide. Thank you very much for your patient hearing.
Yes, thank you for the very informative and interesting presentation. One first question, yeah. More questions? No, I have a short one. And when in the beginning you mentioned one effect, anti-aging effect. How you define anti-aging effect? You have a mice model or just a
So this is the only compound which is allowed to have anti-aging clinical trials, and the dose which is allowed is 2000 milligrams. Okay, so for anti-aging is 2 pills, 1000 milligram, 2 pills.
okay. If the okay, thank you again for this presentation, and I guess we close the afternoon session. Thank all speakers for contributing, discussion, and presenting, and I hand over to the.
Thank you, Professor Fritzl. Thank you for chairing this session. Thank you, Dr. Fadwa. Thank you, all the speakers. And I'm sure that looking at your eyes, that most of you are a little bit tired, but we want to take you for a very short, small tour to the museum, in which you can have just a glimpse of the history of Jordan and some traditional, the traditional museum, archaeological museum. This will take only half an hour to meet then at the restaurant, in which we will be having some music to be followed by the dinner. So thank you for bearing with us for this long day. And let's enjoy the remaining 2 hours.